All righty, let's get started. We have a short schedule, which always makes things scary. Today is our last lecture, finishing off everything we can about the integumentary system. And that's going to be it for this test. It's a quick section, a lot of material, but not a lot of time. So we are going to finish that off. And if we have time, we'll do some more histology at the end. And all leading up to Monday. So the good news is we got a lot of material to cover today, but then you have 5 days to master that material going into Monday's lab and lecture exam. One last quick note, hopefully you figured out at this point, it is always important to be here and be active in the learning class process, being a part of the class. But it is going to be especially true on Wednesday after the exam when we come back. Everybody who is present in class will be formed into groups. And you will be assigned some bones and bone features that you're going to be doing group presentations on. Again, I don't want you to worry about it. I want you focusing on the exam. I know when I say the word presentation, everybody kind of freaks out, but trust me, this is the most informal, laid back presentation you're ever going to have to give. So it's not a big deal. It's not anything to worry too much about, but you do need to be here so that you can be put in a group. You can be assigned a bone and bone feature. You can organize with your group what it is that you're going to be doing. So make sure you are present on Wednesday to be able to participate in that. All right, questions on any of that before we get started? All right, you guys know how much I love my stunned silences first thing in the morning, so awesome. Excellent, all right. So if we have time, if we have time at the end, we will. We got a lot of material to cover, so hopefully we'll have time at the end. We'll see how things go. All right. As we mentioned at the end of last class, we are going to work through the layers of our epidermis. Again, that keratinized stratified squamous epithelial tissue, that 99% of the cells that we see here are keratinocytes. And we also talked about how when we're going to discuss them, we're going to start from the bottom and work our way up. Wow, that line is tiny. We're going to start at the bottom and work our way up because that is the way the cells grow. Of this layer, and again, vocabulary is always important, the layer is the epidermis. There are 4 or 5 distinct sublayers that make them up. And as we talked about last time, they're the stratum basale or stratum germinativum, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. So let's take a look at those. Starting first, and we can start here by just taking this line. Oh, that's a little too big now. And highlighting this single layer of cuboidal and columnar cells that are right on the basal surface of our epidermis. That tissue layer, sublayer, I should say, is what is known as the stratum basale. Basal, of course, referring to the bottom layer, as we know, or the stratum germinativum, right? To germinate means to give birth to, right? Or to sprout from. And so this is the layer where our skin starts the growth process. It is made up of that very single layer of keratinocytes. These are typically cuboidal or columnar in shape. And as the name would indicate, they basically have one function. These are the unipotent stem cells that are going to rapidly divide to produce all the millions of cells that we need to replace the millions of cells that we lose during the course of a day. If you remember, one of the things we know about epithelial tissues is that they're avascular. They do not have their own direct blood supply. That means they can only get their oxygen and nutrients from diffusion, which makes sense that this would be the most active layer, because if you think about it, down here in the dermis, we could have our capillaries, which would then be able to provide the oxygen and the nutrients necessary via diffusion to this single layer of cells that is going to be rapidly dividing, producing the new cells that will start the growth process. Now, remember also down here in our deepest layer is where we said we would find those other random cells that can be found in the epidermis, the melanocytes, the Langerhans cells, and the Merkel cells. What was the function of the melanocytes again?
Melanin.
Make melanin.
Melanin, absolutely, provide the pigment for the skin. What did the Langerhans cells do? Or remember, we can also call them dendritic cells.
Are they immune response?
Yeah, they're the immune cells contain the histamine. They would give us our redness and our rash, help to provide some protection. And what did the Merkel cells do? Or remember, we also called them
Tactile cells?
exactly. They are tactile cells. And what do they do?
They provide the fine
Exactly.
ridges.
That fine discrimination, right? That fine discrimination so that we're able to feel the ridges on a
Sensory response.
Exactly. Yes. But again, remember, sensory means a lot of things: pain, tickle, itch, temperature, right? All of those things are tactile sensations. In this case, it's that fine discrimination, that ability to feel the ridges on a quarter. Remember, as we mentioned, you would not be responsible for identifying a Langerhans cell or a tactile cell histologically. But notice also, real quickly, what type of skin are we looking at the illustration here?
Like a foot or a palm. Foot.
That is where you would find it, but that doesn't tell me what type of skin it is. What type of skin is this?
Oh, it's keratin.
Thick skin. This is thick skin, absolutely.
Oh.
Right? Whereas this one here, what would this be?
Thin.
Thin skin. This is thin skin, absolutely. We'll be able to tell the difference by the number of layers, but also, as the names indicate, we can tell by the size of it as well. All right. So here in our thin skin, notice again, we see plenty of keratinocytes, but notice here in our thin skin, we see the one other type of cell that you would be responsible for histologically, and that is the melanocytes. And how are we going to be able to tell the melanocytes under the microscope?
There's a artifact or free space because they send them to the cells to cover them.
Exactly. It is going to have a very clear cytoplasm to it because you're right. It's making that melanin granules, but it's giving it to all the cells around it. So it's like we said many, many times, and we'll continue to say, right? Color in histology doesn't matter except when it does. And often one of the main ways it matters is by the lack of color. In this case, that lack of color tells us that this indeed is the melanocyte. Notice also, if you look really closely, it looks like we may have clipped a teeny bit of a melanocyte on this one right here. There's a teeny bit of clear. Again, we may have sliced just the edge off of the apple on that one. This second one I wouldn't use on an exam. It's a little ambiguous, but the first one with the big arrow is plenty obvious. So every other pretty much cell we see here in the epidermis is a keratinocyte, but we have just that 1.5, but really for test purposes, that one melanocyte on this as well. So those are things we can see in that layer. All right, now, when this stratum basal cells, and I'll cheat and put a big block here, when this cell divides to become two cells, what does it do to the cells that are above it?
Pushes them up.
Yeah, it pushes them up and pushes them away, exactly. And so as this newly divided cell leaves the stratum basal, it enters into a region we call the stratum spinosum. So the stratum spinosum will be this area here that I will highlight with my highlighter, not the base layer, but basically all of this stuff here. Notice this is made up of multiple layers, somewhere around 8 to 10 layers of keratinocytes. And notice these keratinocytes are still cuboidal and columnar in shape. These cells are now moving further away from the dermis, further away from the blood supply, further away from the stratum basale. So these keratinocytes are no longer dividing. Notice they all have nuclei, so they're not dead, they're very much alive. They're just not using all of their resources to divide. Remember, as we talked about, dividing requires a tremendous amount of energy and a tremendous amount of resources. And so if you're not using that energy and you're not using those resources to divide, you can use that energy and resources for something else. And what might that something else be? Well, what do cells like to do? Come on, what do cells like to do? They're basically factories for the doing of it.
Proteins.
Yeah, they make proteins. There you go. These cells are going to make proteins. After all, what are they called?
Spinosum?
These are keratinocytes, right? What do keratinocytes do?
Make keratin.
Make keratin, absolutely. However, as it turns out here in the stratum spinosum, they're not making too much keratin. They may make a little bit of keratin, but at this point in the stratum spinosum, they have a different job. They're making proteins to form desmosomes. What's a desmosome again?
It's a cell junction.
Yeah, it's a cell junction, absolutely. And it's one that holds the cells together really, really tightly. Remember, it involves a plaque on the inside of the cell, some transmembrane proteins that are going to anchor it to the other ones, some of the intermediate filaments to anchor it in place. So this cell is going to produce all these desmosomes that are going to attach it to the cell next to it. In fact, this is where this layer gets its name. Notice when the cell is producing all these desmosomes to connect it to the cells around it, it gives this cell a spiny appearance. And so this layer is called the stratum spinosum because it is forming all these desmosomes to help to anchor these cells together. Because remember, our goal of our skin is to provide that protective outer layer, form a barrier between us and the outside world. And so by forming these massive numbers of desmosomes, we're able to tightly pack these cells together and provide some integrity to our skin. All right, questions on that? Great question. So these desmosomes that are forming are primarily forming just between the cells in this layer. However, as these cells continue to expand and grow outward, these desmosomes don't go away. So they're going to continue to anchor and hold the tissues together. Plus, we're going to do other things to hold these cells together as well. So yes, this is where they start. This is where they form. But these desmosomes are going to hold basically all the layers together with the exception of that one single layer at the bottom, the stratum basale. All right, notice from here, we finally go into a layer where the cells start to flatten out. Now, notice we have this very dark, very distinct layer known as the stratum granulosum. Granules, remember, are darkly pigmented vesicles. And again, that is where this layer gets its name. right? It has these very, very darkly pigmented, and I'll cheat and grab this and move it out of the way. You can see by far, this is the darkest, most distinct of all of our sublayers because it has cells that are now finely flattened, and they are very dark, very distinctly filled with these very dark granules. The reason for this is now finally we have these cells that are undergoing their major protein production. And remember, even though they're called keratinocytes, there are really two types of proteins they're making. They're making those keratin bundles made up of the keratohyalin granules. These are the ones that absolutely just love the stains that we use, so it makes the layers really, really dark. And remember, keratin is that big, distinct, hard, fibrous protein that gives the skin its toughness. But remember, we also need the skin to be waterproofed. So this is also where we're going to make those lamellar. Lamellar just means laminated layer granules that waterproof the cells together, fusing them together into a single unit. So tightly stuck together that, as we talked about, when we damage the skin cells still peel off as a continuous layer. So the cells are super active in this layer, making massive amounts of proteins, filling themselves up with keratin, waterproofing, and basically shellacking them to the cells next to them. However, as they reach the end of this layer, something special happens. They fill themselves up so much with keratin that the cell says, you know what? I'm so full of keratin, I don't really need this nucleus or these organelles anymore, and it chucks them. And this cell is now waterproof in this waterproof shell, fusing it to the cells next to it. So is it going to be able to get the oxygen and the nutrients that it needs? Is it going to be able to get rid of wastes? No. So what the cell ends up doing is basically undergoing these processes that cause the cell to die. Now, of course, anatomists have a fancy term for this. What is the fancy term they use for this process where a cell actually undergoes a programmed cell death, where it basically kills itself?
Apoptosis.
Exactly. So as the cells leave this layer, basically the cells undergo apoptosis. So notice, basically from this layer up, all the rest of the cells that we see in this thick skin, which as we can see are dozens, if not hundreds of cell layers, there's not a nucleus anywhere in any of them, because all the rest of the cells in the last two layers are all dead keratinocytes.
So apoptosis is the process of expelling.
Yeah, basically expelling the nucleus and the organelles, and basically they just die and become a dead, flattened bag of keratin. So, yep, all the cells from this point down have all been alive, keratinocytes, but all the ones from above are dead. Correct. So yes, if you have the most, especially like on the palm of your hand, if you have a very, very superficial cut, or again, you take your nail file and you were to rub your nail file on the surface of your hand, or even the back of your hand, as long as you weren't too vigorous on it, you're not going to bleed right away, right? You wouldn't start bleeding until you got all the way down to the dermis. If you're able to get all the way down to the dermis, then you're going to bleed. But How far that is depends on the skin we were talking about. Like I said, in the palms of your hands, you have dozens to hundreds of layers of dead keratinocytes. On the back of your hand, you may only have 20 layers. So you have to work a lot longer with that nail file to dig down to bleed on the palm of your hand, but it's a lot easier to do it on the back of your hand. All right, so as we mentioned, from here up, all the keratinocytes are flat, are dead, and fused together. In thick skin, as we mentioned, we have a special layer here directly above the stratum granulosum, known as the stratum lucidum. Lucid means clear. I know this particular layer doesn't look clear, but a lot of it has to do with the stain. Let's cheat and look at this picture. This is the previous picture from your textbook. You can kind of see why they changed it now, because the picture they used for the textbook happens to have a sweat gland going through it, disturbing the layers, which was kind of silly. But what's nice about this is you can see where the stratum lucidum gets its name. Notice it is this very, very clear layer of cells right above the stratum granulosum. Now, let's think about this for a minute. This green layer, the stratum lucidum, which is only found in thick skin, is dead keratinocytes. All of these cells up here that form the stratum corneum are dead keratinocytes. Is there really any difference in the structure or the function of these two layers of the epidermis?
Yeah, one has hair.
No, you're talking about the hair. You're right. Skin itself is different in these two areas, but these two sublayers, is there really anything different about them?
No.
No, there really isn't. And so there's some people that think this stratum lucidum is an artifact. It's not really there. However, every stain we've used shows this distinct layer. So while we don't know why this layer is present in the thick skin, it is something that is indeed consistently found. And since it is consistently found in thick skin, we still identify it. It's dead keratinocytes. It's providing protection. It doesn't do anything different than the layer ahead of it. But it is something that is consistently found in our thick skin. And so it is something that we absolutely positively identify. All right. In thin skin, you would go straight from the granulosum to the stratum corneum, whereas in thick skin, we have this other layer in between. So let's finish off by talking about the stratum corneum. The stratum corneum average is about 20 to 30 layers of dead, flattened keratinocytes. Right, basically all of this stuff up here. Now, is that average number necessarily meaningful? It's like the average height. If I were to give you the average height of a male or the average height of a female, in a class like this where there's 25 of us sitting here, do you think any of us are exactly the average height of the male or female? Probably not, right? If maybe one would be exactly. The people will be close, there'll be a range, but like a lot of things, average isn't as meaningful because there's a huge range. While this is an average, this is a huge range. We kind of hinted at this before, but let's see if we can figure it out. Where do you think you have the thickest stratum corneum?
Feet.
Be more specific.
Soles of your feet.
Close. Heel. There we go. The heel of the foot. Right. That's where you spend the most time with your pumice stone, right? On that heel of the foot. Hundreds, multiple hundreds of dead thickened keratinocytes on the heel of your foot. And where do you think your thinnest stratum corneum is?
Eyelid?
Yep, exactly. The eyelid. Perfect. The eyelid, it is 2 to 3 cell layers thick. So yeah, not just around the eyes, but specifically the eyelid is by far the most delicate of our skins, with only a couple layers of dead keratinocytes in that stratum corneum. So the range is absolutely huge. Great question. Your red margins, what we think of as the lips, it is thin, which is why we're able to see the blood vessels underneath, but it's not as thin as the eyelids. Yeah, the reason we're able to see the redness of our lips is because we have a thin stratum corneum there. And so, yeah, the blood vessels are closer to the surface and we're able to see that redness of the blood in those blood vessels. So it is thin, but the eyelid's even thinner. All righty. Now, the other aspect that I want to talk about this is while we have this thick stratum corneum, if we continue to put force on it, put stress on that, it can actually cause those attachments to come together even tighter. You may not have thought of it in those terms, but you were aware of it from that summer you spent digging ditches, right? Because after spending a summer digging ditches, what happened to your hand?
Callus.
Calluses.
calluses.
Those calluses that form is from the pressure making those attachments even stronger, providing that additional protection. And that's a good thing, because what about when the opposite happens? What about when you're wearing those new shoes and you go for a 5-mile hike and those new shoes are constantly rubbing against the skin? And as they rub against the skin, the layers of our epidermis, or the epidermis and the dermis, separate from each other. And
Get a
that
blister.
area fills with space. Absolutely, you get a blister as a result of that. That blister is a good reminder that while these are dead keratinocytes, while this tissue is hardened, waterproof tissue, it is not completely dehydrated. We do have interstitial fluid in our skin. And that's exactly what happens. When the layers separate, that interstitial fluid accumulates in that area, and we get a blister. Notice also that when that blister forms here in the epidermis somewhere, it is filled with interstitial fluid and it is clear. However, if that separation of the layers occurs between the dermis and the epidermis, the dermis has blood vessels in it. Exactly, that dermis has blood in it, blood vessels in it, and so that space will fill up with blood, and that is that blood blister. So notice a blood blister is typically deeper because it has to involve the dermis, whereas those other blisters are more superficial, just filled with that clear interstitial fluid. Excellent. Questions on that? Bruising is what happens when I don't listen to my wife. Oh, did you want more than that? Bruising is what happens with bruising is that you get the breaking of blood vessels that occur down in the dermis. And so you get an accumulation of blood in that area. And that accumulation of blood in that area can be seen through the surface of the skin. Right, so again, normally it starts a darker purplish color, and then as our phagocytes and stuff break it down, it tends to turn more orangish or even yellowish. It's really not that different from what we want to do, right? Mother's Day is right around the corner. Okay, I guess not, but we could pretend Mother's Day was right around the corner. Mother's Day was right around the corner, as I do every year for Mom to show her how much I love her. I use that permanent magic marker to write "I love Mom" on the surface of my arm, right here on my shoulder, right? Big heart, big mom, permanent marker, so that it stays there forever. And does it really stay there forever? No, even if I promise not to wash it, as we know, eventually those skin cells are going to slough off. So if I want that big heart with Mom to stay forever, what do I have to do with that pigment?
get to the dermis?
Yeah, I have to take it
Tattoo.
and take it all the way down to a less metabolically active part of the skin, down into the dermis. And when that pigment is in the dermis, then again, those cells aren't nearly as metabolically active, and so it stays for a much longer period of time. Notice your cells do start to eat away at that pigment, not as fast as they do with the blood for a bruise, for instance. And different pigments last for a different amount of times. Lighter pigments like yellows and stuff like that tend to be broken down more rapidly, whereas the blacks and blues tend to last for a longer period of time. So they will eventually start to fade in time. These cells aren't dead. They are active. They're just not nearly as metabolically active as we have with our epidermis. So just a tattoo is kind of like a bruise. You have a pigment caused by something in the dermis that you're able to see through the surface of the epidermis. All right, we looked at the actual pictures. I like this picture because it's a nice, pretty illustration that shows all of the same things. Notice again, we have that single layer of cells that is the stratum basale. Notice, conveniently enough, they've given us a tactile cell, that Merkel disc. They've given us a melanocyte. And remember, it can float around the different areas. We have a dendritic cell in here as well. From there, we go into the multiple layers of the stratum spinosum. Notice they have all these spiny attachments from the desmosomes holding them together, gives that layer its name. From there, the cells flatten out and go into major protein synthesis, which is that forming of that keratin. And remember, once they fill themselves with keratin, the cells die. This process of going from newly divided up to the point where it fills with keratin and dies is called the process of keratinization. And this process of keratinization takes around 2 weeks. That dead keratinocyte travels through the stratum lucidum if it's thin skin, or just through the stratum corneum if it's thin skin, and sloughs off. And that process, once the cell dies to the point where it sloughs off and becomes dust in your house, also takes around 2 weeks. So if you think about it, basically every 4 weeks, every month, you have a completely new epidermis. Now, that being said, if I get that paper cut, or if I'm riding my bike and I scrape the surface of my skin, does it take a month for that superficial wound to heal?
No.
No. And the reason for that is when the tissue is irritated, and let's make sure we write this out. When the tissue is irritated, it produces a hormone-like protein called epidermal growth factor. An epidermal growth factor stimulates these stratum basale cells to divide more rapidly. Notice I said irritated. Obviously, if I'm riding my bike and I fall and I scrape my elbow, that is an irritation, that is an injury that can occur. However, shaving can irritate the skin, right? When I use my loofah to scrape off all those dead skin cells because I want to have shiny, smooth skin, I'm irritating the skin. And so when I'm using a loofah to get rid of those dead skin cells, guess what happens? My skin makes cells more quickly. This epidermal growth factor also affects hair. So when I shave, when I wax, when I use a chemical remover like Nair, all of those things irritate the skin, cause epidermal growth factor to be produced, which means that not only does my skin grow back faster, but my hair grows back faster as well. which again, you may not have thought of it in those terms, but you're aware of it because the more you shave, the more you have to shave, right? So again, irritating that skin increases that growth process. Yes, from injury, but anything that irritates the skin causes it as well.
So you know how they say like you do that and then it makes it thicker? Is that anything like an explanation for that? Or no?
It's appearance. When you, the hairs tend to taper. However, when you shave, you cut it to a flat, blunt end. So when it starts to grow back, rather than having that thin, narrow, tapered end at the start, you have that blunt end that is growing out from there. So it gives the appearance of being more coarse, being larger, where rarely it's just you've cut off the small tip and it's the same size it was before. It's not any different from people say that your nails and skin continue to grow after you die. Right, because they talk about how a dead body will, you know, if it sits around for a couple hours, will have a 5 o'clock shadow, right? You know, where the hair will occur. It's not that their hair is continuing to grow. What actually happens is their skin starts to dehydrate. And as their skin starts to dehydrate and shrinks away, the hairs that were near the surface as the skin shrinks away suddenly become apparent. Right, so the nails aren't getting longer, the hairs aren't getting longer, the skin is shrinking, and so it appears like those things are happening, but they're not actually happening. All right.
I have a question.
Yes.
What part of the skin, like if you have like acne, but then it scars and you get like that dark pigmentation, what part of the skin does that stay in? Because sometimes I know people that have had scars for years. Does that happen in the dermis?
Yes, exactly. The scarring that occurs happens in the dermis. And in fact, when we will talk at the end of class today about the two main healing processes, regeneration and fibrosis, and how that fibrosis occurs in the dermis, and that is what causes the scarring to occur. So yes, if an infection becomes large enough, or if it becomes irritated enough, or you pick at it enough, or something along those lines, scar tissue can form as a result of that, yes. Hold on, I saw there was another question, but I switched screens for a minute because there was one other thing, important, very, very important thing that I wanted to show you on this. So that's there, but let's come back and see what the question was. No, having the hair grow faster isn't necessarily a bad thing at all. Absolutely. Again, washing the skin is important. However, again, like anything, all things in moderation, right? We kind of already talked about this before, but actually we'll talk about it more. I guess it was the other class I was talking about it. How many of you have ever worked in the food industry before? Although now, for most of us, again, when COVID first came out and everybody talked about how important it was to wash your hands and everything, and people were washing their hands 15 times a day. When you wash your hands 15 times a day, what starts to happen to your skin?
It dries out.
Dries out. Why does it dry out?
You're removing the oil?
Yeah. Well, we'll use the term oil for the next half an hour until at least we talk about what that oil absolutely is. But you're absolutely right. There's an oily secretion that it helps to maintain the integrity of our skin. And there is resident bacteria, right? A normal centimeter of your skin has something on the order of 10,000 bacteria on it. And I know when I say that, everybody goes diving for their Purell, but those bacteria are a good thing. They are benign. They are out competing harmful bacteria, so that if a harmful bacteria tries to land, the bacteria that's already there is eating all the resources and using all the resources, and that bad pathogen, that bad bacteria can't find a purchase. So if you're constantly washing, then you're removing those oils, you're removing those bacteria, those helpful bacteria, and that can be potentially damaging. So like with all things, there's moderation. Yes, it is important to wash. Not washing can affect bacterial, can cause acne, can cause things along those lines. So like anything, everything in moderation. All right. Now, the other thing that I wanted to show you is we have looked at this, and you are absolutely positively responsible for these things histologically. However, even though we haven't been in the classroom, there are a lot of really excellent resources in the classroom. And one of those, under our modules, in the lab handouts, you will see there are two pictures of two skin models that are in the classroom that I will already tell you right now with 100% certainty are going to be on the exam. Notice there's some great things that we can see on this. I'll use my highlighter to show this. Notice we can see that it shows both thick and thin skin. Notice we can see the living cell layers. They don't really do a good job of distinguishing the stratum basale from the stratum spinosum, but we know the stratum basale is going to be the deepest layer. So that's that part right there. And the rest of it is the stratum spinosum. We then have a decent stratum granulosum in here. And notice here in yellow, we have the stratum lucidum that we don't have in the thin skin, and a much thicker stratum corneum than in the thin skin there. So we see those types of things really, really nicely. And notice there is a second one that does an even better job of showing this. Notice here, again, we can see thin skin and thick skin. Notice here, stratum basale, stratum spinosum, stratum granulosum, stratum corneum. So 4 layers in our thin skin, and basal, spinosum, granulosum, lucidum, and a much thicker corneum in the thick skin. So these models do an excellent job of showing us our epidermis and the sublayers associated with that layer that is the epidermis. But notice also, if we go back to the previous picture, which I think does a better job of showing the relative size, notice even in our thick skin, this is the epidermis, and all of this is the dermis, right? This is the epidermis, and all of this is the dermis. So if in this tiny little layer, we can fit 5 sublayers, how many sublayers do we put in the dermis? What do you think?
1.
1 would be nice, but no, it's not quite that nice, but it's 2. So it's not too bad. It is just 2. There are 2 sublayers to the dermis. All right, so any other questions on the epidermis before we move down to the dermis? And actually, let's go back to that picture. And I think I'll use the other one. So, all of this is the dermis, right here, from here to here. Someone remind me again, how many different connective tissues make up the dermis? 2. How many sublayers did I say there were in the dermis?
2.
2. Do you think that's a coincidence? No,
No.
it's not. Their first layer, the superficial layer, basically making up about 20% of the dermis, is the region that is known as the papillary layer. So superficial, 20%. Papillary layer. Oops. And it is the papillary layer that is comprised of our areolar connective tissue. The remaining portion of our dermis, making up about 80% of our dermis, is what is known as the reticular layer. And of course, with a name like the reticular layer, What type of connective tissue is it comprised of?
Reticular.
Reticular would make sense, but absolutely we know that's not true. What's the other connective tissue that makes up the dermis that we already learned?
Irregular connective tissue.
Yeah, dense irregular. So if it's made up of dense irregular connective tissue, and there is such a thing as a reticular connective tissue, why do they call it the reticular connective tissue? When are you guys going to learn the easy answers? There you go. Mitch has got it because anatomists hate you. Now, actually, reticular means met or a net or mesh-like. And again, the dense irregular connective tissue has this mesh-like structure. So, I mean, I guess there technically is a reason for it, but it is definitely confusing. All right, let's look at these layers in the lecture. The papillary layer is named the papillary layer because of its formation. Notice every time we've looked at the dermis and the epidermis, let's just cheat, there's our skin. Is the dermis and epidermis just this flat layer that sits on top of this other flat layer? Is that what the dermis and epidermis has looked like in any of the pictures or histology slides we've looked at?
No.
No, there's all these undulations. And that's what papillae means. Papillae is a finger-like extension. So notice our dermis has these finger-like extensions that stick up and interlock with our epidermis. Now, why would that be useful? Why might we want to have those interlocking finger-like connections between the dermis and the epidermis?
For strength.
Yeah, absolutely. Right? If you put your two hands flat against each other, and even if you push really hard, you can still slide them back and forth. Is that what you want happening to your dermis and your epidermis?
No.
No. But if you interlock your fingers, then your hands aren't going anywhere. And remember the other thing we know about our epidermis. The other thing we know about our epidermis is it's avascular. So it has to get its oxygen and the nutrients from the dermis. And so by having all these undulations, it gives us an increased surface area. And that increased surface area is better for diffusion. So it locks the cell layers together better, and it gives better diffusion. So this areolar layer, this papillary layer is made of the areolar connective tissue, and it helps to hold the cells together, the tissue layers, together, and it gives us better diffusion. Now remember, areolar is a loose connective tissue with a lot of space. So that means in these papillae is space for a lot of stuff. Not surprisingly, one of the stuff we have are capillary loops. This is where we're providing the oxygen and the nutrients, removing the wastes for the epithelial tissue that is the epidermis. But there are sensory structures here as well. Notice here on our illustration, right here in one of these papillae, we see this kind of elongated tube or pill-shaped structure. This pill-shaped structure located in the papillae is what is known as a tactile corpuscle. Now, it's also known as Messner's corpuscle. And of course, why is it called Messner's corpuscle?
Because you found it.
You found it?
Yeah, good old Bob Messner was the first one who identified it and described it and planted his flag in it. And again, if you're related to Messner, then that's a useful piece of information. But I also like the term tactile corpuscle a lot because that sounds an awful lot like tactile cell. And what did our tactile cells do?
The fine Find, what's the word? I can't think of it.
Discrimination.
Discrimination.
Absolutely. And these tactile corpuscles also provide fine discrimination. So they give us the exact same type of sensory information as the tactile cell, but there are two big differences. The first is obviously the sublayer where we find them. Tactile cells are found in the stratum basale, whereas tactile corpuscles are found in the papillae of the dermis. But the other big difference are the regions of the body where we find the most of them. Where did we find the most tactile cells?
In the palms of our hands, our fingertips.
Yeah, palms of our hands. Where else?
Fingertips.
True, palms of our hands, especially the thumb, fingertips, but there was another location as
The
well.
mouth.
Face. Exactly, face, especially lips. Tactile corpuscles are found in the largest papillae. And as it turns out, the largest papillae are located on the palms of the hands and the soles of the feet. If you think about that, what do we know about the skin on the palms of the hands and the soles of our feet? What type of skin do we find in those areas?
Thick.
These are found more in thick skin. In fact, notice if we go back to our model of the skin, we do not see and will not be tested on tactile cells, but we know the tactile cell would be sitting here, hanging out here in the stratum basale of the epidermis. But notice what we do see here in the thick skin, that pill-shaped structure located in a papillae. So guess what this right here is? It's not a trick question. What were we just talking about? There you go.
The tactile
corpuscle.
corpuscle.
Exactly. And do you think our other model has it too?
Yes.
Does it?
No.
No?
Maybe.
Yeah.
Oh.
Notice in our thick skin, we have these pill-shaped sensory structures.
That's
We
really
have
sad.
our tactile corpuscles. Nope, I forgot one. I missed one. There we go. Perfect. Excellent. Now, tactile corpuscles are not the only sensory structures we see here. And let's cheat and go back to the picture for a second. Let's see which one's better. I like the other one. We see more of the model. So notice here and here, and some of them are coming. I don't actually have it in this one. So that one's. We have some of these nerves that are coming up and just ending. These nerves with no special ending as their name would indicate, are free nerve endings. And these free nerve endings provide us with general sensory information. By general sensory information, I mean things like itch, things like tickle, things like pain, and things like temperature. Temperature is one of the tricky ones, right? Can you dip your finger in water and tell, oh, hey, that's exactly 98.3 degrees Fahrenheit?
No.
No. right? But if you were in elementary school, when you were learning about your five senses, one of the games they have kids do is they have 3 bowls of water. One is warm water, one is room temperature water, and one is cold water. And they put one hand in the cold water and they put one hand in the warm water, and the kids let them sit in there for a minute. And then they take both hands and put it into the room temperature water. All right, if you never experienced this, then do this after class today. Right? Warm, not scalding hot water. But if you do that, when you put both hands into the room temperature water, does the room temperature water feel the same to both hands?
No.
No.
No. We don't actually measure absolute temperature. We actually measure, or we perceive, temperature changes. So the hand that went from warm to room temperature feels cold. The one that went cold, room temperature, feels warm. We measure temperature changes. And again, if you think about this, you knew of this. If you're sitting in a hot tub and I were to come and turn it up 1 degree, would you be able to feel that change? No. Now, let's take that a step further. Does that mean if every 15 minutes I turned it up 1 degree, if you stayed in that hot tub long enough, could I boil the water with you not being aware of it?
No.
No.
No. Well, for one, you'd have to stay in there for a very, very long time. But it turns out the answer is no. And the reason for that is we actually have a second type of temp receptor that is actually a pain receptor. So basically what happens is you wouldn't feel the gradual change. However, you do have a critical temperature receptor. And when the temperature reached that point, you would feel pain. And when you felt pain, you would get out. But guess what? Not everything has that second temperature receptor. Like a frog doesn't. Now, I'm not encouraging any of you to do this, but you could put a frog in an open pan of water, and every 15 minutes, you could turn it up 1 degree. And if you were to do that, you could actually boil the water and the frog would never jump out. The frog would never notice the change in temperature. The frog doesn't have that pain receptor for the temperature. And so you could actually boil the water and that frog would never jump out. Not necessarily. Again, temperature we perceive as changes, as I mentioned. Typically, when really cold, like if you're doing one of those cryo treatments or something like that, or you're burning yourself with scalding water or something along those lines, you're right. Those things are more pain types of information. And as far as I know, cold and hot pain don't move any differently. So the speed of those should be similar. I would think it's the other way around. I would think we would measure hot faster because like fire and things like that that are burning you. But maybe it's something different about water because colder temperatures are further from our water. So it's a larger temperature change. So maybe something like that would make it more perceivable because it's more, because to get from body temperature to freezing versus body temperature to boiling. No, actually boiling is still higher. I don't have I have not heard of that. I'm not saying that it's not true. I'm saying I'm not aware of the neural pathway by which something like that would occur. So I won't say no, but I'm not aware of that. All right. Excellent. So, that was the other, sorry, I wanted to go back to that. Those free nerve endings actually can be found in the papillary layer, but they also can be found in the epidermis as well. So we can perceive temperature, we can perceive pain in both the dermis and the epidermis. I love this picture from your textbook. Again, it shows using an electron microscope. It shows that areolar connective tissue and how there's all these fibers, but there's lots of room for all sorts of other stuff, blood vessels and mast cells and all sorts of other hunk and junk hanging out there in the papillary layer. Now notice, as we talked about, the papillary layer is more enhanced in our thick skin region. And you may not have thought of it in those terms, but you're aware of it because when we get wet, those papillae become enhanced. So if you're in that hot tub for 2 hours, what happens to you?
Your fingers raisin.
Yeah, you get pruny, you get raisins. But do you turn into raisins all over? If you look at your
No.
skin, at your
Only in
forearm,
our-
does it get pruny?
Only on our thick skin.
No, only in our thick skin. And the reason for that is that in our thick skin, these papillae become so large and so enhanced that they actually form ridges in the epidermis underneath them. Again, you may not have thought of epidermal ridges before, but you're probably aware of it because it's what we commonly refer to as our fingerprints. Of course, as you know, we don't just have fingerprints. We also have palm prints and footprints and toe prints and heel prints. Everywhere that there is thick skin, we have these enhanced ridges. They help with our grip. Like I said, when we're wet, they become enhanced to help us to grip things that are wet. For the most part, these things are genetically determined. Now, I did say for the most part, so what does that mean?
Not always.
Not always.
Not all of it. So do identical twins have identical fingerprints?
No.
No. Very similar, much more similar to each other than two non-identical siblings would have, but they're not identical. And what's remarkable about our fingerprints is they actually form in utero. So those two identical babies with the identical genes in mommy's tummy, there's something unique about that uterine environment. It could be a difference in concentration gradients, a difference in temperature, a difference in the surface of the uterine wall. There's something about that environment that causes a modification so that everybody has a unique genetically determined, mostly genetically determined, but a unique pattern to these ridges. And again, you've got an activity that you're going to be doing or did do, where hopefully you took a look at some of these minutiae of details or what are called Galton points. And I guarantee you'll be asked about those on the lab exam. So hopefully you did that activity or at least looked at it. All right. The remaining 80% of our dermis, as I mentioned, is the reticular layer. That reticular layer is made up of dense, irregular connective tissue. Again, here's what it looks like under the microscope. And see, indeed, we have bundles of collagen and elastic fibers going at all sorts of different orientations. Again, remember the fancy term we used for this. This provides protection from multi-axial stress. So no matter how we rub the skin, we're provided some protection. But it's still a connective tissue, so there's still space. And as we look at this picture, we can see more hunk and junk that's found in here, including the hair follicles. Hair follicles are the structures that contain, support, and grow the hairs. So we'll talk about the follicle and the hairs a little later today. We have our oil-producing glands, what are appropriately referred to as sebaceous glands, also associated with that hair follicle. We have our sweat glands, and again, we're not going to get away with terms like sweat and oil glands. We're going to need to use the appropriate anatomical terms, sebaceous and sudoriferous glands. But there's also some sensory structures down here. Notice in particular, we have this one right here. this onion-shaped structure. Notice it has many layers to it, which again is where it gets its name. It's a lamellated corpuscle. Of course, it's also called a Pacinian corpuscle because good old Bob Pacinian was the first one who identified it and described it. And the way these onions work is when these layers are squeezed, it stretches the membranes out and they send a signal to the brain. So basically, these multi-layered structures form what perceive as pressure.
Great, there's a lot of background noise.
Sure, I can mute everybody and then you guys can unmute as you need to talk. These laminated corpuscles are what allow us to perceive pressure. So if someone grabs your arm and squeezes, or you squeeze a tennis ball, or the feel of your chair on your back and on your butt as you're sitting in your chair, that sensation of pressure is from the squeezing of these lamellated corpuscles. And then, of course, also, since we have the tiny capillaries in our papillary layer, we have our large blood vessels and our nerves down here in the reticular layer. All right. Questions on that? All righty. That's it. That is our skin. Now, the one last thing I want to remind you, well, actually, there's two more things that I want to tell you about. Notice how when we looked at this electron microscopy view, using an electron microscope, we see that collagen fibers project in all sorts of directions for that multi-axial stress. But are all parts of our skin equally stressed in all directions? Especially someplace like my elbow. Is my elbow just as equally likely to be stretched up and down as it is left and right? No.
No.
So not surprisingly, there is actually a general pattern to the way that our collagen fibers are aligned. Notice here we have this nice illustration that shows this general pattern of how the collagen fibers are aligned. And these are what are known as the cleavage or the tension lines. So notice we go back to that elbow we were talking about. Notice you're much more likely to stress the elbow up and down as you're moving it and stretching the muscle than you are left and right. So it gives us an important indication of the general functions of the skin and how they're going to be stressed. But this is also clinically significant, right? If I'm going to do some kind of surgery on this individual's leg, is it going to be better if I make my incision with the lines of cleavage or against the lines of cleavage?
With.
With is going to have much less scarring, much less damage, and will heal much more rapidly. In fact, if you think about it, if you look at it, even in this simple illustration, you can see how elaborate the lines of cleavage are in the face region. And that's one of the major parts of plastic surgery. One of the major components of plastic surgery is to know how to make those incisions where you're going to minimize the scarring so you can make those improvements with the minimal amount of damage and the most rapid healing as possible. All right, the last thing I want to mention about this stuff is, again, to emphasize that hypodermis. Remember, the hypodermis is associated with the skin. It is adipose and areolar connective tissue that loosely connects it to the muscle underneath. But again, remember, it's not a part of the skin. The skin is just the epidermis and the dermis. And so we have talked about now the main organ that is the skin, So all we have left to do now are those accessory structures, the hairs, the glands, and again, to emphasize some of the sensory things that we need to talk about. This is a good stopping point for our first break. So let's go ahead and take our first break. It is 9:20. That means we will restart at 9:35. So any questions, and I'll start the recording. Any questions on the dermis or epidermis before we switch gears? All right, excellent. Then I will see you guys in 15 minutes. All right. So let's go ahead and get started. We have been talking about the skin, and now we are going to talk about the accessory structures, the glands, the hair, the nails, and then sum up our physiology. As I mentioned, when we talk about our glands, for all glands, we obviously need to know the name of the glands. We need to know the substance they produce. And probably along those lines, probably is not a bad idea to understand what the function of that substance is, know why we secrete it, right? But remember, we also talked about how we need to know the structural classification. We need to know the functional classification. And for the skin, it's probably good to know the region or regions of the body where you would find it. So let's think about all of these things while we're talking about our glands. Maybe if I make this a little smaller, I can sneak it up here in the top and we can keep this here. We'll start easy. The first gland that we are going to be responsible for is what is known as a sebaceous gland. It's the one that commonly makes what we think of as the oil of our skin, but do not use oil gland on the exam. You won't even get partial credit for that. For full credit, you need to say sebaceous glands. When we talk about the regions of the body where sebaceous glands are found, they're found over the entire surface of the body, except for the palms of the hands and the soles of the feet. And what is special about those regions?
They're hairless.
Exactly. There's no hair in those regions. So they're absent in the palms of the hands, the soles of the feet. Or the other way we can think of these sebaceous glands is that sebaceous glands are also associated with hair. So in fact, they release their secretion into the follicle of a hair. So without a hair, you can't, or at least a hair follicle, right? Some of us have follicles with no hairs on them, in them, but we still have those follicles. And without those follicles, you cannot produce and release that sebum. Here, we see a simple illustration of how that works, where we have this sebaceous gland connected to a follicle, so that that oily secretion, that sebum it produces, is released out of that gland into the follicle and then out from that follicle to the surface of the skin. Oops, I can't erase that because I need to keep this up at the top of the screen. As I mentioned, we need to know the structural classifications and the functional classifications of our gland. For a sebaceous gland, they are all branched acinar or branched alveolar. And someone remind me what that means? If only I had learned about the ducts and the secretory structures and how they're put together to make a gland structurally.
Is that 2 or more ducts?
2 or more ducts?
Glands.
2 or more secretory structures. Oops, that's not ready. Excellent. What else do we know about this gland? Acinar means circular shape. Absolutely. The secretory structures are circular shape. And it tells us one more thing about it. What's that one more thing? Nope, doesn't have to do with the elastic and the collagen fibers.
What it secretes?
No, remember, when we talk about the structural classification, we determine that by the shapes of the secretory structures, the number of secretory structures, and the number of ducts. We know a branched gland has 2 or more secretory structures, but how many ducts does a branched gland have?
1.
Yeah. Exactly. So when I tell you that a sebaceous gland is a branched acinar gland, these are the 3 things that it tells you. That tells you that it has 2 or more circular shaped secretory structures connected to just 1 duct. And that's actually what we see really nicely in this illustration. In fact, this illustration has 2 things that help us to very, very easily identify, really 3, to easily identify that we are looking at a sebaceous gland. The first absolute dead giveaway is this follicle. This structure right here is the hair follicle. This would be where the hair is located. Now notice the hair isn't here anymore, but this follicle is a very distinct shape that is easy to identify, and our gland is connected to it. So the fact that our gland is connected to a hair follicle is a dead giveaway that we are looking at a sebaceous gland. But there's a couple other really important things that we can see here as well. What's another color that'll show up really well on this? Maybe the yellow, maybe? Let's see if this works. Notice we just have a single duct coming out into that, and these big, large, ball-shaped secretory structures. Now, the last thing that I want you to look at is notice the cells. These cells are big and huge and bulbous. In some ways, they look a little bit like adipocytes. Adipocytes are really big and bulbous and clear because what do adipocytes contain inside of them?
Fat.
Fats, absolutely. And this is an oil-producing gland, so it has a lot of fat as well. However, notice with the adipocytes, remember the nucleus is pushed to the side. Is the nucleus pushed to the side inside of these cells?
No.
No.
Oh.
So it may try to trick us into thinking this is adipose, but when we see those nuclei in the center of the cell, that tells me that these are the oil-producing cells producing not oil, but sebum, the oily secretion that is being released into the hair follicle. So by identifying those 3 things, the hair follicle, the shape, and the color of these secretory structures, I can easily identify this as a sebaceous gland. All right. Now, if you remember, we talked about these sebaceous glands once before, and we said that they use the holocrine mode of secretion. Remind me again what that means. How many days does it test again?
The cell bursts.
Yeah, exactly. It fills with the secretion and then bursts. It ruptures and dies, releasing all of its contents, producing an incredibly thick and incredibly viscous secretion called sebum. And what is the function of this sebum?
To keep.
Hydration?
Yeah.
Well, so, yeah, okay, it helps to moisturize the skin, maintain the skin to keep it from drying out, Absolutely. And the hairs as well. So the hairs don't become brittle. The skin doesn't become dry and cracked. There's actually a minor antibacterial component to it, where it kind of helps to make the skin a little bit more acidic to prevent harmful things from, bacteria and stuff from coming in. Remember, we don't want to wipe out all bacteria, just bad bacteria. So it has these important functions to it. So notice, just that simply, we have answered all of the possible questions we need to know about our sebaceous glands. All right. Name is sebaceous. What is the substance it produces?
Sebum.
Sebum. What's the function of that secretion?
Holocrine.
No. So what is the function of the sebum?
It moisturizes and waterproofs.
Yeah, and has an antibacterial component. What is the structural classification of our gland?
Branchicular.
Or remember, we could also say branched alveolar. Remember, those two terms are interchangeable. What is the functional classification of the gland?
Holocrine.
Holocrine. And where is a region in the body you would find it?
Your arm, your hair.
Yeah,
your head.
arm, any part of the body that has hair on it. So brachial region, antebrachial region, scalp, right? Buccal region, right? At least for me, right? Femoral region. All of those regions are locations, basically anywhere except the palms of the hands or the soles of the feet. All right. We get how the game is played?
Yes.
Excellent. And remember, as we talked about, this holocrine mode of secretion produces a very thick, very organic material. And with only one duct, that duct can become congested with that material. It becomes congested with that material, and yet it's very warm, it's very moist, it's very, very organic, just the kind of things that bacteria like. So bacterial can get in, can infect that congested gland, producing acne, right? And these glands become more active at puberty. So the hormones at puberty cause these glands to be more active, meaning they're more likely to get congested, meaning you're more likely to have acne during puberty, or times of stress, or other things that can cause that increase in glandular secretion. All right. Let's do the same thing with our sweat glands. Now, there are actually 2 plus 2 major types of sweat glands. Well, isn't that 4 types? Kind of. 2 of these are actually glands that produce sweat. And 2 of these are glands that are modified sweat glands and produce something very, very special. Now, the good news is that all 4 of these glands all share the same structural and functional classifications. So I'll write it up here so that we remember all sudoriferous glands. I'm going to make this smaller. From a structural standpoint, are simple, coiled, tubular glands. And what does that tell us? What does it mean to be a simple, coiled, tubular gland?
Things can get in and out easier, like
So remember, when we talk about epithelial tissues, simple epithelial tissues, simple means one layer of cells. But when we talked about glands, remember it meant something else. Remember when we talked about structural classification, which again, I guarantee is one of the essay questions in your test bank that you could be responsible for. When we talk about the structural classifications of glands, it is the number of secretory structures, the shape of the secretory structures, and the number of ducts. So a simple coiled tubular gland has one duct. How many secretory structures? One secretory structure. And what shape is that secretory structure?
Tube.
And what does the coiled mean?
It's a coiled tube.
Yeah, it's twisted. It's not in a straight line. It's not in one big long straight line. It's twisted up. Notice they've done a nice job of showing this to us here on our illustration. Notice here they've given us this nice three-dimensional structure of how we would have a single coiled tube. However, when we look at these things under the microscope, is that how we see it? Are we able to see the entire three-dimensional structure of that coiled tube?
No.
No. So notice what they've also done a nice job of doing for us here histologically is to show us that when you actually cut through a tube like this, what you get are a bunch of pieces of that tube. Some of them are cut straight on, some of them are cut oblique, some of them are cut longitudinally. So we get all those irregular shape tubular structures. So that is what it looks like under a microscope because it is a simple coiled tubular gland. One duct, one tubular secretory structure that is twisted up. Now, functionally, all pseudoriferous tubes, all pseudoriferous glands are the same as well. They all use the merocrine mode of secretion. And remind me again what that was.
That's when the cell isn't damaged.
Exactly. The cell is not damaged. Instead, the cell simply uses, like Mitch pointed out, exocytosis. It puts it in a vesicle, brings it to the plasma membrane, and releases its contents. And all pseudoriferous glands use the merocrine mode of secretion. It is vitally important to remember this fact. Because what's the other vitally important fact you're supposed to remember? That anatomists hate you. And this is going to be one of the places where we see it. Let's talk about the two types of sweat glands. The first type of sweat gland is what is known as an eccrine sweat gland. This is by far the most common type. And every once in a while, you will see someone refer to an eccrine gland as a merocrine gland. Now, of course, why would they call it a merocrine gland?
Because that's what its function is.
Yeah, well, that's the functional classification. That's how it releases its secretion. But that's how all pseudoriferous glands release their secretion. So is it smart to just take one of the four types and name it after the mode of secretion that all four of them use?
No.
No.
No, that's incredibly stupid. But sometimes you'll see it. Eccrine glands are the ones that we see here. Notice this is the histology of this one here. Notice a couple of key things. Notice, as we've talked about, we're going to see a bunch of irregularly shaped tubes, some cut and cross sections, some oblique, some longitudinal. As soon as you see all these clusters of tubes together, the really important thing to look at is the size of the lumen. What do you notice about the size of the lumen versus the size of the wall for this particular gland? Which is
Do they
bigger?
all vary?
Well, they do vary a little bit, but which is typically bigger, the size of the hole in the center or the size of the cells making up the wall?
The cells.
Cells.
Yep, there you go. Exactly. So notice this one has a very small lumen. And the reason it has a small lumen is because these eccrine glands produce our watery sweat. These are the ones that help us to regulate our body temperature. Now notice I said it's the most common because what part of your skin sweats?
A lot of places.
Yeah, in fact, let's switch the question around. Is there a region of your body that is covered with skin that doesn't sweat?
Yes.
Where?
No, I don't. I sweat everywhere.
Yeah, exactly. Absolutely. So if on the exam, I asked you to identify a region of the body where you would find these eccrine glands, is there a wrong answer?
No.
No.
No, because these things are everywhere.
We wrote like everywhere. Would that be wrong?
Technically, yes, because that's not a region of the body. Right, so technically that would be wrong. Now, I likely wouldn't mark it wrong, but technically in the wording of the question, that would be wrong. So just give me one region and you'll be fine.
Okay.
Now, does that mean they're equally distributed over all parts of your body? No, of course not. You have more of them, for instance, on your palm of your hand than you do on your forearm and stuff like that, but they're pretty much everywhere. In a nice, cool, controlled environment, like when you're stuck in Zoom classes for 8 hours a day, sitting in your air-conditioned house, they produce about ½ liter of sweat during the course of the day. Of course, instead, if you're spending 8 hours of a Sacramento summer tarring a roof, are you only producing 500 milliliters of sweat during the course of that?
No.
No.
No. In a 24-hour period of time, these glands can produce as much as 8 liters of sweat.
Ooh.
Right now, do you have 8 liters of extra fluid in your body that you can afford to get rid of in a 24-hour period of time?
No.
No.
No. It's so vitally important to hydrate when you are sweating. Absolutely. And of course, because it's not just water you're losing either. You're also losing antibodies and ions and things along those lines as well. And like we talked about, alcohol, wastes, garlic, all of those things as well. All right, now take one more good look at this eccrine gland. And let's compare that to our second type. Let me cheat and do some erasing here without getting rid of the stuff on the top that's important. And talk about our second major type of sweat gland, what is known as an epicrine gland. If you remember, I think I mentioned these to you guys once before, where they used to think that these glands use the epicrine mode of secretion. Of course, we now know they don't. They use the merocrine mode of secretion, but they didn't change the name. These epicrine glands, remember, as we talked about, are some of the fewest found in the skin, only about 2,000 total, and they're not randomly distributed. They are associated with hair, but in particular with secondary hairs. What do I mean by secondary hairs? Anybody know what I mean when I say secondary hairs? Come on, I know you. There you go. Liz has got it. These are hairs that become present after puberty, right? One of the big ways we can tell the difference between a boy and a man, a girl and a woman, is the presence of these secondary hairs, these hairs that become active and present at puberty. Where would you find these? Give me a region of the body.
Genital.
Yeah,
Axillary.
absolutely. The pubic region. Where else?
Axillary.
Axillary region, absolutely. Thoracic, buccal, right? All of these areas where we get those secondary hairs. These produce a sweat that is more of an organic sweat. We talked about this before. Again, this is not about temperature regulation. We don't have hormones per se, but the smell of this sweat when it is first produced is something that most people find pleasing. It has a nice musky kind of aroma to it. However, being organic, it is a tremendous amount of more viscous organic materials within it, which is why they thought it used the upper mode of secretion, and that's why they named it that. And bacteria love that organic sweat. So that's why that t-shirt that you worked out in and then you left in your gym bag for 3 days, or my teenager who won't wash for 2 weeks straight, right, start to get that nice pungent aroma that we associate with body odor because of the bacteria feeding off of that organic material. So notice here with our illustration, they've done a nice job of showing this gland connecting to a hair follicle. But notice also if we go back, oh no, didn't want to do that. Here we go. Notice again, we see a simple coiled tubular gland. We see a longitudinal, a cross section, an oblique section. But this time, notice the difference between the size of the cells making up the wall and the size of the lumen. What do you notice about the size of the lumen here?
Huge.
Huge, much, much bigger. And that's because instead of producing a very watery sweat, this is producing a very organic sweat. So we need that bigger lumen for the more organic sweat. And that's the way under the microscope, we're going to easily tell these two apart. All right. Questions on that? All right, now notice I mentioned there are two other types of sudoriferous glands that are highly, highly specialized. You may not be aware of the fact that they're sudoriferous glands, but I know you're familiar with them, so let's talk about them. The first is what is known as a ceruminous gland. Now again, like all sudoriferous glands, it is a simple coiled tubular gland. Like all sudoriferous glands, it uses the merocrine mode of secretion. But anyone have any idea where this one is found and what it does? I'll give you a big hint. If you look closely at the picture, especially right here, it tells you where this is located. What is the external auditory meatus?
Is that your ear?
Yeah, exactly. It's your ear hole.
So it produces earwax.
Exactly. It produces earwax. Of course, are we going to get away with a term like earwax on the exam?
No.
No. So it actually produces cerumen. So our ceruminous gland produces cerumen, but cerumen is earwax. You are 100% absolutely correct, right? What does cerumen do?
Protects the
Ear. How?
ear.
How does it protect the ear? You're 100% correct. How does it protect the ear? Jackie
It-
got it too.
Collects dirt and bacteria and stuff, so it doesn't get to your
Exactly.
eardrum.
It captures dust and debris to stop things from trying to get inside. And it actually has a little bit of an insect repellent quality to it. Now, that doesn't mean that the next time you go camping, you can just rub earwax on your arms and the mosquitoes won't get you. But is it important to discourage bugs from going into your ear holes?
Yes.
Yeah, absolutely. And so that cerumen helps with that as well. Now, I want you to notice one more thing. Notice when you look at this under the microscope, it has a big, huge, massive lumen, just like our epicrine sweat glands. So can we easily tell these two apart under the microscope?
No.
No. So I'm not going to show you this gland under the microscope. You won't have to know this gland histologically. You won't have to recognize it under the microscope. And the same thing is true for the second one. The second specialized sweat gland are the mammary glands. Where are those located?
In the breasts.
In the breasts, absolutely. And what is their function?
Produce milk.
Produce milk. There you go. So 4 types of sudoriferous glands. All are simple coiled tubular glands. All of them use the merocrine mode of secretion. But all 4 are found in different locations and have different functions. All righty.
Question.
Yes.
So for the location, we wouldn't be able to say ear, so we would say like what, the otic region?
You could say ear canal. I
Or
would say
we can say ear. Okay, and we
You
can
would
say
say ear.
breast for the mammary glands.
Well, that's because it is your breast.
Yeah, okay.
All right. And this is your ear canal. Those are correct anatomical terms.
Okay.
All righty. Excellent. So there are our glands. Bingo. From here, we need to talk hair. Yeah, mammary glands produce milk. When mature, I should say. Males, a female who has never been pregnant, they both have mammary glands as well, but those mammary glands are immature. They're not capable of producing milk. However, when a female becomes pregnant, she produces hormones that matures those glands. And once they're mature, they become capable of producing milk. So yes, technically, everybody has mammary glands, but not everybody produces milk. All right. All right, let's talk about the hair, and let's do this in a simplified version first. Here is our skin. Here's our dermis and our epidermis. And hanging out inside of here is our hair. So we'll put a hair in like this. Hold on, let's do it this way. So there is our hair in our skin. And when we talk about the hair, our hair has, no, too small. Three regions, and it also has 3 layers. Let's talk about the layers first. And sometimes it's easier to see the layers if we actually were to take a hair, and let's actually cheat here. Make my skin a little shorter. And make a circle. Our hair, like many structures we're going to see in the body, are composed of an inner region and an outer region. This is a type of organization we'll see in the kidney, in the adrenal glands, in the lymph node, in the ovary, we're in bones. We're going to see this type of organization again and again and again. And when we have this type of organization, the inner part is referred to as the medulla, and the outer part is referred to as the cortex. And so not surprising, the hair, like many of the structures of the body, have that type of an organization. So again, at the center of our hair, we have a medulla, and then we have a cortex. Again, very, very common type of organization that we see a lot. However, one of the special things about the hair, especially out here on the outer surface, actually, let's do this, where it's exposed on the outer surface out here, it needs a little extra protection. So what happens is our hair has this fuzzy layer of very thick overlapping keratinized cells on its outer surface. This heavily keratinized outer layer is what is known as the cuticle. So when we talk about the three layers of our hair, from deep to superficial, we have the medulla, we have the cortex, and we have the cuticle. All right, you with me so far? Now, stop me if you've heard this story before. We have a single layer of cells that divide rapidly. And as these single layers of cells divide rapidly, they push the cells above them up. As the cells move away from that dividing point, they fill up with keratin, fill up with so much keratin that they end up dying and become dead, flattened keratinocytes fused together.
Yes.
Right? Exactly. The exact same way our hair grows is the exact same way our skin grows. We start producing cells and pushing them up. In fact, if you also think about it, there are also melanocytes in this layer producing pigment. And what does that pigment do?
Hair color.
Gives you your hair color. One of the interesting things about these melanocytes is, unlike the ones in your skin, in many people, and it is almost primarily genetically determined, some of these melanocytes will stop producing pigment. And when they stop producing pigment, what happens to that hair?
Turns gray.
Yeah. Do you get that gray hair or that white hair as a result of it? Absolutely. So we start with these keratinocytes that then continue to develop and mature. Part of that maturation process is the formation of this cuticle. There is almost no cuticle present when the hair is first produced, but as the hair continues to grow, the cuticle gets bigger and bigger until finally it reaches a fully intact cuticle state. So notice, if you think about it, when we talk about the regions of the hair, we have three regions to the hair. We have the place where the growth takes place, and that is known as the bulb. We have the part where the cuticle is forming, and this is the root. And then we have the part where the cuticle is fully formed, and this we call the shaft. So when we have the three regions from deep to superficial, we have bulb, we have root, and we have shaft. So notice we have three regions to the hair and also three layers to the hair. Now, there's two more things we have to identify. This single layer of cells where the hair is produced is called the matrix. As we know, the matrix is producing massive numbers of cells every day to allow that hair to continue to grow. But like we said, this is just like the epidermis. Keratinocytes dividing, hardening, dying. And like the epidermis, these cells are avascular. So they don't have a direct blood supply. Luckily for us, there is a finger-like extension of the dermis that sticks out into the bulb, and it contains the capillaries, the blood vessels and the nerves. Wow, that didn't show up at all. I'll use blue then. The blood vessels and the nerves that are going to be in here to help, or the blood vessels to provide the nutrients and remove the waste for this to grow. And this finger-like extension, as we know, a finger-like extension is a papillae. So this is the hair papilla. Papilla singular, papillae plural. So in the bulb, we also have a papillae that is going to help it to grow. You may not have thought of these things in these terms, but you are aware of it. Because as we were talking beforehand, as we know, your overall beauty is directly related to the amount of hair you have, especially if you're a female. So one of the things that a female may decide to do is to shave. When she shaves, she takes that razor and rubs it across the surface of the skin, removing the superficial part of the shaft that is showing. Then you get that nice smooth skin. You can be accepted by society. But what's the downside of that?
It causes it to grow more rapidly.
True. Part of it, as we talked about, it causes that epidermal growth factor to grow more rapidly. But notice also, by just removing the superficial layer, the rest of the hair is right there under the surface. And how long does it take for that hair to start poking out again?
Not long.
Not long at all, right? So maybe instead, you may want to do something like a chemical removal like Nair or waxing. When you wax and yank on the hair, when you use that chemical removal like that Nair, basically what ends up happening is that it tears or dissolves the hair down here where the cuticle is not fully complete. So as a result of that, we remove a larger portion of the hair. And of course, what's the advantage of that?
It
Takes longer to grow back.
Takes longer to grow back, but it will still eventually grow back. So if I want that hair to never grow back, what can I do?
Get a laser treatment.
Laser is one of the ways they do it. They used to use a process called electrolysis, but you absolutely have the right idea. If we want to eliminate this hair forever, what we actually have to do is destroy the matrix. Now, let's go back to that fun process of electrolysis. Again, all of you are far too young to remember electrolysis, but maybe your mom or your grandma might even have had this process. What they would do is take a needle, and basically they would insert this needle into the follicle of the hair, all the way down to the bottom where the matrix was. That line is not thick enough. All the way down to where the matrix was. They would then produce an electrical current down at the tip of that needle and hopefully destroy that matrix. And if you destroy that matrix completely, the hair stops growing back. Of course, if you don't completely destroy the matrix, what happens?
It grows back.
It will eventually grow back. And can you tell whether you've completely destroyed it or not?
After a while, yeah.
After a long time, but not right away. So take a quick look down at your forearm. Count how many hairs you have on your forearm.
A lot.
A lot, absolutely. And they had to go one hair at a time with those needles to try to destroy that matrix. Does that sound like a fun and quick process?
No.
No, it was incredibly excruciatingly painful, incredibly long process, and you weren't even guaranteed with 100% certainty that it would work. The things we do to be beautiful, right? Now the process is a little better. As you guys mentioned, we can use lasers. We can penetrate the skin with those lasers and hope to destroy those cells at the matrix with that laser. Again, you're not always going to get 100% of it. Some of them may grow back, so it may take multiple sessions. But the nice thing about the lasers is it's less painful and you can do it over a larger surface of the skin at one time. So it's a little bit faster, a little bit less painful. But like I said, it's all worth it as long as you're beautiful on the outside, because that's all anybody cares about. What's inside doesn't matter. It's all what's on the outside that counts. All right. Hopefully you guys get sarcasm over a Zoom. It's hard to tell sometimes. All right. Questions on yes.
Yeah. I know this is probably like off topic because we won't really be covering it, but what about ingrown hairs? Like what happens with that?
What happens with an ingrown hair is typically, and we'll actually kind of get to that in a second, the hair grows outside of the follicle. The follicle, as we're going to see, is the support structure around the hair. But if the hair penetrates through the follicle, it can actually grow into the dermis or into the epidermis and cause problems. So that's what an ingrown hair is. It's when the hair doesn't follow that follicle out. All right. Which is actually an excellent lead into what we need to talk about next. So any questions on the hair before we move on to the follicle? Here, in fact, let's look at the pretty pictures from your textbook that show this. Notice here, we see the hair, and notice this picture shows the medulla, shows the cortex, shows the cuticle. We see the papillae in there in the bulb region. Although I think this is one of my favorite regions. Here we go. The three regions, as we mentioned, we have the bulb where the hair grows, the root where the cuticle is forming, and then the shaft where the cuticle is fully formed. This is the picture that I like. Hold on, there we go. Notice this is again an electron microscopy view of a hair as it is coming out of the skin. Right, you pull that hair out. I won't do that because I only have 5, so I don't want to pull them out. But if you were to pull a hair out and look at it, it looks smooth, right? But when we look at it under electron microscope, we see that what that cuticle is these massive thick layers of overlapping keratinized cells, providing tremendous protection. And that's important for the integrity of the hair, because if the cuticle becomes damaged, what happens to the hair?
It doesn't grow back.
No, it isn't so much that it doesn't grow back, but when the cuticle gets damaged, what happens? The hair loses its integrity and the hair can
Split.
Yeah, you get those split ends. Split ends are caused by damage to the cuticle. All right. So that is our hair. But notice also the hair is surrounded by a follicle as well. This is the sheath that contains the hair. And this helps to maintain the strength and the integrity of the skin. Notice if the hair was just poking out through a hole in your skin, And when you look at how many hairs are on your forearm, if those were just holes in your skin wouldn't be very protective. So notice what actually happens is the epidermal cells actually extend down and wrap around the hair, maintaining the integrity of the skin and providing that protection. And in fact, it's not just one layer of epithelial cells, it's two. Notice when we look at the pretty picture here, here again are those layers we were talking about for our skin. Here is the cuticle, here, pardon me, the medulla. Here is the cortex. Here is the medulla. Oh God, I did it backwards again. Medulla, cortex, cuticle. The outer layer is the cuticle. Notice here in yellow and here in purple are not one, but two layers of epithelial cells. The one closest to the hair is the internal epithelial root sheath. And the one that surrounds it, the thicker one, is the external epithelial root sheath. So notice we go back to our picture that we've been drawing. We will draw an orange layer around the outer surface, which is the internal epithelial root sheath. And then in purple and thicker, we'll draw the external epithelial root sheath. Hold on, I see there's a question here and then someone just raised their hand. Is it the same person? Well, let me do this and then I'll answer the questions. And then let's draw them around here as well. Our internal epithelial root sheath and our external epithelial root sheath. So when we talk about the layers of our follicle, it's going to turn out to have 3 layers as well. Excellent. All right, so let's answer these 2 questions. Why do people say if you pull out your gray hairs, more grows back? It is totally a myth. And again, there is absolutely no logic behind that. My guess is that if you're at the point where you're nervous about it and pulling hairs, then you're probably more aware of them, so that you may be more aware of them as they're growing out. But again, we know they turn gray because these melanocytes. Now, why the melanocytes stop producing melanin, that we don't know. We don't understand why that occurs, but we do know that there's a very, very strong correlation. So again, if your mother went gray early, you're likely to go gray early if you're female. If your dad went gray early, you're more likely to go gray if you're a male. So there is a strong genetic component to that. But the whole pulling a hair and having 3 gray ones grow back doesn't work, because if you think about it, if that was the case, then I'd be pulling hairs left and right. I don't care what color they would be, just as long as they came through, but that's not how it works. So we don't have to worry about that. Aubrey, what was your question?
For it to be 2 epithelial tissues, does that mean that there's a free space in between?
That's a great question. And no, the answer to that is no. There is no free space in between them. So they're kind of like multiple layers almost, or sublayers, the way they are. But you do bring out an amazing point that I wanted to make next. And that amazing point is, as we have talked about before, yes, there needs to be a free space above them where the hair sits. But remember, also on the basal surface, we have a basement membrane. And what's remarkable about these hair follicles is these hair follicles are one of the only places in the body where we might actually see that basement membrane. Not every stain's going to show it all the time, but there are some where we're going to be able to see it. And if we're going to be able to see it, then of course, anatomists are going to give it a name. And what they call it is a glassy membrane. So some of our hair follicles, on our hair follicles, we will actually see this thin layer of glassy membrane between the epithelial tissues and the connective tissue. And that's the last thing. Obviously, if these are epithelial tissues, there's no blood supply, so to get nutrients and things along those lines. So we need an outer layer, and that outer layer is what is known as the dermal or connective tissue root sheath. So we have the glassy membrane and the dermal or connective tissue root sheath. So 3 layers to a hair, let's call it 3.5 layers to our follicle. And again, my picture here has gotten a little messy, so let's look at the pretty pictures from your textbook. Notice here, we see this a lot nicer, where we again see the internal epithelial root sheath, the external epithelial root sheath, our glassy membrane, and then our connective tissue root sheath. And it does really look like that. Oops, hold on, let's cheat. Where were we looking at the, oh, I know where we were looking at it. We were looking at it with the glands. So let's go all the way back. Perfect, perfect. Now, obviously, we don't have the hair in here. If we had the hair in here, we would see that the hair has a medulla, the hair has a cortex, and here inside the follicle, it might have a little bit of a cuticle. So we'd see the hair there. However, notice if we look at the follicle, we can clearly see the layers of the follicle. Here we see the internal epithelial root sheath. Here we see the external epithelial root sheath. And notice, I'll change to a highlighter so I don't hide it too much. Notice there is this clear layer that wraps around it. This clear layer that wraps around it is our glassy membrane. Remember we said if you use that glue right, you never see it. But this is the one place in this class where we'll actually see the basement membrane. And because we see it here, we give it a fancy name, the glassy membrane. And then that is surrounded on the outside by our dermal or connective tissue root sheath. So we can see the 3.5, 4 layers, if it makes you happy, of that follicle here. So absolutely positively, you're responsible for this histologically. Oh yeah. Not this one, but actually, let's actually go back for a second. This is a good summary picture. Notice if you look closely at this model, when we were talking about it before, we saw that very clear distinction between our thick skin and our thin skin. But notice our thin skin also is divided into 2 parts. And on this particular model, this would be thin skin, for instance, from the back of the hand or the back of the arm or the scalp, whereas this would be the axillary region. What's the difference between the axillary region and the scalp region of our thin skin? I don't know either, so we won't bother. Well, let's think in terms of glands. What type of gland is this one here? Identify the specific gland.
So the simple coiled.
That is its structural classification, but that's not the type. What type of gland are these two glands?
Wet or pseudophorus?
You get partial credit for sudoriferous, but I said specific. Eccrine, excellent. And where should eccrine glands be located?
Everywhere?
Everywhere. So notice our thick skin, our air armpit, and the top of our head all have eccrine sweat glands. Notice no hair in our thick skin, but the scalp and our axillary region have hair. So notice they have these, what I'm highlighting, these bulbous purple ones connected to the hair. What might that
Sebaceous.
be? Sebaceous glands, excellent. But notice there's a third gland here.
It's the
Notice it's a
apical.
simple coiled tubular gland, but with a much larger lumen, and it happens to be connected to a hair and in this axillary region. So what's that one?
The apicrine.
Epicrine.
Apicrine.
So notice, or again, I don't care how you say it as long as you spell it correctly. But notice with these three regions, it shows us those differences. The other model is divided into three parts as well. Thick skin, thin skin, thin skin. Again, this would be the scalp. Again, this would be the axillary region. And again, notice we have that apocrine gland, very obvious here in the axillary region and not in the scalp region. Okay, but that's not actually why I'm here. We've been talking about the hair and its follicle. And notice if you look closely, this model shows us all 7 of those layers. We've been going from deep to superficial, so let's go the other way. Layer C. What layer is layer C?
Cuticle?
Dermal connective tissue sheath.
It is the dermal root sheath of the follicle. Excellent. What would D be then? Glassy membrane. Perfect. What does that make E?
The external epithelial sheath.
Perfect. F?
The internal
Internal.
epithelial sheath.
Excellent. G?
Cuticle.
Yep. Now, technically, should the cuticle be this complete, this close to the bulb? No. But again, our artist is doing a nice job of showing this. What's H?
Cortex.
Cortex. And what's I, this little white part in here?
Medulla.
Medulla. And notice they also even give us our dermal papillae in there as well. So notice our model also does a beautiful job of showing us the hair layers and the hair and the follicle layers. And of course, I could also do the regions on the exam. If I pointed here and asked you to identify the region, what region would that be?
Scalp.
No, not the region of the hair.
Oh,
Region of the hair is that.
Shaft.
Shaft, excellent. Whereas down here, what would it be?
The root.
And then down here, what would it be?
Bulb.
There you go. Layers and regions, hair and follicle. Just that easy. Questions on that? All right, let's quickly do the nail, which is going to be even easier, assuming we can get all the way back to it. Excellent. Stop me if you've heard this story before. We start with a single layer of cells known as a matrix that divide rapidly and push the cells up above them. As the cells get pushed away, they harden, fill with keratin, fuse, and die till they get to the end where you chew them off. The same way your skin grows, the same way your hair grows is the exact same way your nail grows. You produce keratinocytes, they fill with keratin, they fuse together, forming a hard structure. We talked about the function of the glands. We talked about the function of the hairs. What's the function of the nails? What do nails do for you?
Is it still used for protection?
Mhm.
Just because.
Protection, right?
Yeah.
Scratch someone's eyes out, pop the top on your soda can, right? Grasping, gripping, pinching, right? Protection. Absolutely, all of those are kind of things. So again, we know the basic structure, we know the basic function. Let's talk about the anatomy. As I mentioned, we start with the nail matrix where this grows. If the nail becomes damaged, it can fall off. When I was in 3rd grade, I was living back in St. Louis at the time. And in St. Louis, they have these magical things called basements, which is a room underneath your living room. And we have another magical thing in St. Louis called snow. So it gets cold outside. So you have fireplaces. I was carrying some logs up from our basement up backstairs to bring them to our fireplace, tripped, fall, log fell on my thumb, damaged my thumb, squeezed my thumb. I lost my thumbnail, but my matrix was intact. So what happened?
Grew back.
It grew back. If you damage the matrix, does it grow back?
No,
No. Did that happen to anybody?
not me. No.
No. My wife's best friend has twins. And one time when she was putting those twins in the car, she closed the door, sliding door of the minivan to put the kids inside. And one of the kids had left their hand outside and she closed it on her pinky, smashing her pinky finger in the door, damaging the matrix, and her pinky nail never grew back. Of course, the nice thing is now she can easily tell the twins apart, but you damage that matrix and the nail no longer grows. We need the nail anchored in place, and there are 2 main anchor points. The first is what we commonly call the cuticle, but you will not use that term on the exam. And that is a thin fold of skin known as the eponychium or eponychium. It also has to be anchored distally. This is a region we commonly think of as the nail bed, but you're not going to use that term on the exam. You will use the term hyponychium. And I don't like that. So don't use cuticle and nail bed, use eponychium and hyponychium. As I mentioned, the rough edge that you file or you chew on is what we call the free edge of the nail. The bulk of the nail is the nail body. But notice underneath the nail, where the matrix starts, there is a big, thick region of epithelial tissue. This big, thick region of epithelial tissue, notice, squeezes most of the connective tissue out of the way. And so as a result of that, that small area of the nail where there are fewer blood vessels, it is whiter in color. That area that is whiter in color is known as the lunula because it kind of is a half moon shape. In fact, when you look at the nail, it has that color to it, and that color is there because of blood. If you don't believe me, grab your thumb and squeeze it, and when you look at it, it will appear white, and then you'll see the red flow back into the area. Now, again, this happened in the classroom, but I still need to give a warning. I had a student who was very panicked because when she squeezed her nail, the color didn't change. And so she was very upset and concerned about that. And it turned out her nails were painted. So I'll warn you right now, if your nails are painted and you squeezed your nail, you will not see a color change. All right, but if it isn't painted, then when you squeeze that nail, you will see the color change. All right, questions on that?
I have a question.
Yes.
How come some of your fingers have more of a lunula than others?
It has to do with the shape of the bones, the shape of the finger, and that shape of the matrix and underneath it. So all of those things, the smaller ones tend to have less blood vessels in the area, so they can be a little bit bigger relatively. The thumb is an area where you have a much thicker skin layer, a much thicker matrix, because the nail of your thumb is a little bit thicker. So those are things. So there are a couple factors that can cause variations in the size of the lanella. Yep. All right. The good news is we are done with the anatomy. We still have some physiology, but the good news is most of this physiology are things that we've already talked about and we're going to summarize, or some that we've kind of pointed at and we will emphasize them a little bit more. So let's come at this information fresh. It is 10:45, so we will restart at 11. If you haven't done so already, I encourage you to get some caffeine, right? Get a coffee, get a Coke, do a bump of cocaine, whatever it is you need to get through the last hour and a half, because that time is going to fly. We got a lot of material to cover. All right. And I will start the recording at that point. Any questions? No. Also helps if I spell recording right. All right. See you guys in 15 minutes. And don't do cocaine. It's bad for you. All right. We are in the home stretch. Let's talk about some of the physiology. Again, a lot of this is going to be review and things will be adding layers too. Let's start easy. Skin color. What is the primary determinant of skin color?
Genetics?
Say again?
Genetics or melanin?
Okay, yeah, exactly. Melanin. You got absolutely the right idea. The pigment melanin. But you are also correct, and it is the genetics that affects that. Because as we mentioned, all of us have approximately the same number of melanocytes. The biggest difference is both in the amounts and in the colors of the melanin that we produce. Anywhere between yellow reddish to a brownish black. And as I mentioned, there are at least 4 genes involved in determining that coloration. And then, of course, aside from the genetics, we also know that UV radiation plays a huge role in how much of that melanin we produce as well. While melanin is one of the primary changes, it isn't the only, right? This does allow changes, but does this allow dynamic changes in skin color? Are you like able to walk up next to a very dark tree and you can instantly change the color of your skin to blend into it?
No.
No. So while it can change skin color, it's a slower process. Along those same lines is a second pigmentation known as carotene. Carotene is actually a precursor to vitamin A. So when we ingest that carotene, our body is able to convert it into vitamin A. And what color, based on the name, do you think carotene is?
Orange.
Yeah. And how do we get this orange pigmentation?
Carrots.
Yeah, primarily from our diet. Absolutely. Our diet, not just carrots, but also like yellow and orange bell peppers and things along those lines, have this yellow orange pigment in it that can be stored in our stratum corneum. And so the superficial layers of the skin, and also the adipose below the skin. Why is this carotene important? Why is vitamin A important? Anyone know?
Doesn't that help eyesight?
Exactly. It is vital for healthy eyesight, which of course is why you never see a rabbit wearing glasses, right? They eat lots of carrots, they have good eyesight. Right? Again, this primarily comes from your diet. So it's not a major factor in skin color, but your diet, vegetarian versus more carnivorish, can have an effect on skin color. Where you really see this is in individuals who start juicing. Jack LaLanne is a famous example. He was a person who was famous for juicing, did a massive amount of juicing. And when you're juicing massive amounts of these orange and yellow pigmented vegetables and then drinking them, his skin took on a nice orangish coloration. I had a student, gosh, it was a summer school. It was close to 10 years ago now. where she had an 8-year-old son who had decided that he was eating nothing but carrots. And after 3 weeks of that, his skin took on a nice orangish glow to it as well. So this is one, again, affected by our diet, but again, not dynamic changes. Can our skin color change dynamically? Well, let's think about this. If, as I mentioned, we are giving group presentations, you'll be doing it on Zoom, so it's not quite as bad as when we're in the classroom. But if you were in the classroom giving a presentation, and while you were giving your presentation, your pants suddenly fell down, might there be a change in your skin color as a result of that?
Yes.
Oh, flushing.
Yes.
Yeah, or you walk up to that individual at the bar and use your cheesiest pickup line and they slap you across the chest. I mean, across the face. By the time you get back to your table, you have that nice big red handprint on your face. Absolutely. We can have dynamic changes in the coloration of our skin. And those dynamic changes primarily come from our blood. And as we've talked about, that protein hemoglobin is the iron binding oxygen to it. And when that iron binds the oxygen to it, it gives the skin a nice red pigmentation to it. Now, some people's veins are closer to the surface of the skin, so they may have more of a reddish coloration. Others may be deeper, so they may have a whitish or bluish coloration to their skin. So the locations of the blood vessels can vary, affecting the coloration of the skin. But with our blood vessels, we can dilate and constrict them. And as we dilate and constrict them, that changes the amount of blood in the area, which changes the pigmentation. And that's important because can changes in the coloration of the skin give us some indication of a condition that somebody has, the condition and health
Yes.
of their body?
I see.
Yeah, absolutely. What color is somebody if they're cyanotic?
Blue.
Yeah, there's a bluish coloration to the skin. What causes that bluish coloration?
Lack of oxygen.
Absolutely. Lack of oxygen or lack of blood in the skin. So for instance, when you get cold, your blood vessels constrict to bring the blood to the core of the body. So there's less blood in your skin and it gets a cooler color. Or, as you mentioned in a more extreme example, not enough oxygen. So it could be an indication that someone's got a plastic bag over your head. Right, so that change in coloration of the skin, that bluish coloration, can be an indication of poor body temperature or poor oxygen levels. Conversely, what color is the skin if the skin is erythemic?
Red.
Yeah, that's a red. This red is caused by congestion of blood in the area. Probably the best example is that one that I used when you're embarrassed, or when you get slapped across the face, or when you get sunburned. The next time your significant other has their back sunburned, what I want you to do is take your hand, come up and press it very firmly on their back. When you do that, what you will see when you remove your hand is you will see a white print of your hand in that location. That redness from the sunburn is from a congestion of blood in that area. So when you push on that area, the blood goes away, it becomes whitish in color, and then the red will come back. So do it 3 or 4 times. I'm sure your significant other won't mind that you're pushing on their sunburn. I just explained to them it's for science. All right. So changes in blood flow to an area can tell us something about the condition. But there are other things that can change the color of the blood as well. What color is your skin if you are jaundice?
Yellow.
Hello.
What causes that yellow coloration?
Your liver.
What is it?
Is it your liver? Like
You absolutely have the
something's
right
wrong.
idea. Right. There's
I
the
don't know.
typical, so no, you are absolutely have the right idea. However, that the liver isn't actually what's responsible for the color, but you're absolutely right. What happens is the skin takes on a yellowish coloration because of a buildup of bilirubin. Bilirubin is a byproduct from the breakdown of red blood cells. As we talked about when we were talking about tissues and blood, red blood cells are formed elements that don't have a nucleus, don't have organelles. A red blood cell only lives for about 100 to 120 days. So every day, millions of our red blood cells are being broken down, and we need to make millions of them to replace it. When we break down the red blood cells, we break down the hemoglobin, and one of the products we get from the breakdown of hemoglobin is bilirubin. Our bilirubin is, as you mentioned, used by the liver to make our bile. What does bile help us do?
Like uses protection in your stomach.
It's not just protection, but it actually, we use it to help to break down fats. And during that digestion of fats, our bilirubin is broken down into urobilin, which is actually reabsorbed by the body and released by our kidneys. So it actually gives the yellow color to our urine. And it is broken down into stercobilin, which gives us the brown color for our feces. So the color of our urine, color of feces, tell us something about the function of our liver. Now, as Eva pointed out, if the liver is not functioning properly, because you've been drinking vodka for breakfast, lunch, and dinner every day while you've been in this COVID quarantine, the liver is not able to break down that bilirubin properly, and bilirubin builds up in our blood. And as it builds up in the blood, we get a yellow coloration that is easier to see when the blood vessels are close to the surface. So like in the conjunctiva of our eye or our lips or our nail beds, these are places where that yellow coloration comes out. Where else besides alcoholics is jaundice very common?
Babies.
Infant.
Babies, absolutely. Newborn babies. Recent studies show as many as 30% of newborn babies show some level of jaundice. It's quite common. And the reason for this is our fetal blood, while we're a fetus growing up inside of mommy, the fetal blood has a massive amount of red blood cells. Huge oxygen carrying capacity because of the massive amount of growth and metabolism that is taking place while that baby is growing. At the time where a baby is born, a large amount of those blood cells die off. And the problem is many babies, their livers aren't fully formed yet. And because they're not fully formed yet, they're not able to fully break down that bilirubin. And so they show some level of jaundice. Now, as I said, this is very typical, where you typically see it rise to a certain point at about day 2 or 3, and then it rapidly declines after that. But if a baby is jaundiced, they will monitor the baby closely. Because if the bilirubin levels go too high or stay too high for too long, bilirubin can actually interfere with neural development. So if it stays too high or stays high for too long, it can be dangerous. So how do they deal with it? Or better yet, how did grandmas deal with it back in the day? If a baby was jaundiced, what would grandma do to it? Come on, I know you know. There we go. Grandma would put the baby in a window. Now, again, not like on the windowsill like a potted plant, right? She'd have a nice diffuse curtain, but she would give her that indirect sunlight, because sunlight contains UV radiation. And that UV radiation breaks the bilirubin down into a more easily digestible form so that the liver is better able to deal with it. Which is why, if you go to any neonatal ICU unit, you'll see they have a huge wall of windows where they put all the babies when they're jaundiced, right? Is that what you find in the NICU? Bunch of windowsills? No, that would be cool, but it's not. What do they have instead?
UV lights?
Yeah, UV lights, UV blankets. So it's the exact same thing, right? Getting that UV radiation to help to break down the bilirubin.
Does
All right.
vitamin D have anything to do with that?
I'm sorry?
Does vitamin D have anything to do with that as well?
No, you're right. Remember, sunlight converts a cholesterol in our skin to vitamin D. But so the UV radiation is useful for that as well. But in this case, no, this is the UV radiation is affecting the bilirubin. So it doesn't affect vitamin D levels, or vitamin D levels and jaundice are not related. Let's just say that. All right, one more easy example. What color is someone's skin when they have carbon monoxide poisoning? No?
I don't know.
Typically, a person will take on a bright pink coloration to them as a result of this. Right. Remember, we talked about we have this hemoglobin, and it is the oxygen that is binding to that iron that gives it the red coloration. Well, the problem is carbon monoxide wants to bind to that exact same place that oxygen does. And the problem with carbon monoxide is it has a stronger binding and doesn't like to let go. So it will bind to the iron and it will not let go. And suddenly your hemoglobin can't carry oxygen anymore, which is a problem because your hemoglobin is responsible for basically transporting about 98% of the oxygen to the cells of your body. So if you fill up your hemoglobin with carbon monoxide, can we get the oxygen that we need to our cells?
No.
No. And while oxygen binding gives it the red color, carbon monoxide, when it binds, tends to give it a pink color. Right? Carbon monoxide is very, very dangerous, and it's scentless, which is why they put that scent in it. So if you smell that, or your detector senses it, or suddenly the people around you are going pink in the face, grab them and yourself, get out into an open, exposed area, and get help immediately, because it is very, very dangerous. All right. Questions on that? I don't think that's too tricky information. Hopefully that makes some semblance of sense. All right, let's switch gears to another important function of the skin. We talked about thermal regulation. The primary way that our skin helps us to regulate temperature is by the movement of blood. Your skin is a blood reservoir. While you're sitting here resting in class, a large amount of your blood is just slowly chugging through your skin, slowly working its way back to your heart. Because of that, we can use that to regulate our body temperature, right? If instead of sitting here in our chairs, we decided to stand up and exercise, what would happen to those blood vessels in your skin?
They would open up.
Yeah, exactly. The blood vessels dilate. That brings more blood to the surface. And why do we bring that blood to the surface? What does that do?
There's that circulation.
Well, it is making the circulation faster, but think of it this way. If your significant other just finished vigorously working out, you look at them and their skin is all red, and you just put your hand near them. You don't even have to touch them. You just put your hand near them. What do you feel?
Heat.
Heat. Exactly. And that's the key. By bringing all that blood to the surface, we can radiate off the excess heat. So by bringing it to the surface, we radiate it off. The heat is just coming off of them in waves because we've brought that to the surface. We'll talk about sweat in a minute. So we'll talk about sweat in a minute, but right now we're just talking about blood flow. Conversely, what happens if we are exposed outside in the cold and our temperature decreases? What happens to our blood vessels then?
They constrict.
Yeah, they constrict. So less blood to the surface. And that does two things. We radiate off less heat, and it keeps the blood at the core of our body where our vital organs are to help to keep them warm longer. That's a good thing for the heart and the lungs and the stomach and the liver, but it's not as good for the skin, especially the real distal parts, like the tip of your nose or your ears or your fingers. When they're exposed to cold for a long period of time and now blood's not going to them, not only are they not getting the oxygen and the nutrients that they need, but they're also not getting the heat that they need as well. And so what happens to those distal structures when you're exposed to that cold for a prolonged period of time?
They turn blue.
Yeah, not only can they turn blue, but even beyond that, what can happen?
They die.
Yeah,
Yeah.
the tissues can actually die. Exactly. The tissues can die. Frostbite. Exactly. We can get frostbite as a, right? Notice you don't get frostbite on your liver. But like I said, those distal structures, your fingers, your toes, your ears, your nose, these are the areas where that blood is not going to, and you can have that problem. So notice this vital and dynamic movement of blood can help us to maintain body temperature. But as you've also pointed out, Augustino, I think looks like pointed out, there are other methods that our skin, remember we're talking about the integumentary system, there are other ways that the skin can help us to regulate body temperature. One of those is our sweat, right? Now, let me get rid of this for a second. When I talk about sweating, we're talking about sensible sweat. And by sensible sweat, I mean the sweat that you can see when you run around the room and you see sweat on your arms. And now we can be more sophisticated. What glands are responsible for that watery sweat that helps us to regulate body temperature by evaporating away heat?
Pseudofurious?
So which ones?
Pseudofurious?
So you're right.
Yeah.
But
Ugh.
do all pseudoriferous glands play a role in regulating body temperature? No,
No.
specifically the eccrine. So while pseudoriferous is a correct answer, notice eccrine is a better answer because it's more specific. And remember, as we talked about, you can produce as much as 8 liters of sweat in a 24-hour period of time. Now, I think I saw someone mentioning that their AC is not working, but hopefully for the rest of you, you're in a relatively controlled environment and you've been sitting down listening to lecture. So as you look at your arm right now, do you see any big beads of sweat on the surface of your arm right now?
No.
No. But guess what? You are losing water right now, even in that controlled environment. It's what we call our insensible sweat. This insensible, well, not sweat, our insensible perspiration is basically just the loss of interstitial fluid through the skin. Remember, as we talked about, our skin is mostly waterproof, but not 100% waterproof. So while you're sitting here, a little bit of your interstitial fluid is evaporating away. Now, is it a massive amount?
No.
No, typically less than 500 milliliters a day. It is typically a very small amount. However, if your skin is damaged, if you have 3-degree burns over large portions of the body and that skin has been damaged, then are we able to lose a lot more water in that situation? We've lost that waterproof barrier of our skin.
Yes.
Yeah, absolutely. And so one of the things, remember, we talked about people who have severe burns, we have to watch out for pathogens getting in, but we also have to worry about losing moisture. So they're typically kept in a sterile and a very humid environment so that they don't evaporate away too much water. Excellent. So we've talked about those. Notice there is another way that our skin, tries to help regulate body temperature, and that is our goosebumps. All right. Our hair has the ability to stand up on end. When does that occur?
We're cold,
When we're cold, or when else?
scared,
Scared.
or nervous.
Absolutely. So why? Well, this is a mechanism that is supposed to help us to maintain body temperature, right? Anybody here ever scuba dived before?
Yes.
Excellent. When you put on your scuba suit, is it like in the James Bond movie so that you can keep your tuxedo underneath perfectly dry?
No.
No, when you put on that scuba suit, what do you actually do?
You're protecting your temperature.
Yeah, you actually put water inside of it, don't you?
Oh yeah, yes.
Yeah, the reason you put water inside of you, as you absolutely mentioned, is to maintain body temperature. If you were just sitting out in the ocean naked, then what would happen is the water of the ocean would come by you, and as it went across the surface of your body, it would steal warmth from your body. Your body would radiate heat into the water, and that conduction of heat would be taken away by the currents. However, that scuba suit keeps water right next to you. So when the water steals your heat, the scuba suit keeps that water right next to you to help you to maintain body temperature, right? Think of Siberian husky dogs. They have these huge, long, massive hairs that stand up in their body when they're cold. And they do the same thing. The wind of Alaska is flying by them and would steal their heat, but their hair captures some of that air, keeps it close to their body, and helps to maintain their warmth. Now, is it as successful for us as it is for that Siberian husky?
No.
You step outside on a winter day to pick up the newspaper and the door locks behind you. And the problem is you're in your pajamas. And the problem is you don't wear pajamas. So there you are naked on the front porch in the middle of winter, but then your hair all stand up on end and you feel warm and comfy.
No.
No, it's the same way with fear. What does your cat do when it gets scared?
The hairs on the back of it.
It arches its
Yeah.
back and its hairs stand up. Why?
To appear larger?
Yeah, so it looks larger and more intimidating, which is exactly what happens with you. You and your significant other are walking down the street after watching a movie. Some bad guy comes up with a gun and asks you for your money. And then your hair all stands up on end and they get intimidated by your size and they run away. Again, obviously, these are things that aren't functional for us in real life, but the evolutionary processes are still there. So when we get cold, we get scared, the hairs stand up on end, but it doesn't play a great job on helping us to regulate body temperature. Right? Now, notice what's missing from this list. What's another way that we regulate body temperature? Yeah, shivering. However, what are you using when you're shivering?
Your muscles?
Muscles. And are muscles part of the skin?
No.
No. Remember, we also talked about our fat helps to provide insulation that helps us to regulate
Shivering
temperature.
is for metabolism, right? So you'll heat up.
Well, we'll talk about that. We'll come back to that question in just a second. So shivering, though, is skeletal muscle, and it is, again, not a part of the skin. Adipose, providing insulation, helps us to regulate body temperature. But is that adipose, that subcutaneous layer, is that part of the skin?
No.
No. So notice there are other ways we regulate our body temperature, but these are the only ones associated with the skin. Now, let's get back to the shaking muscles. The reason your muscle shakes is your muscles are motors. They convert chemical energy into mechanical energy, ATP into movement, just like your car. Your car converts chemical energy, gasoline, into mechanical energy, movement. And when your car drives you to grandma's house 2.5 hours away, you're so excited to be at grandma's house, you give your car a nice big hug and lay your face right on the hood of the car while you're hugging it, right? Yes?
It's way too hot.
Exactly, because motors are not perfect. When they're converting chemical energy into mechanical energy, some of that energy is lost in the form of heat. And our muscles are the same way. In fact, some of our muscles, as much as 75% of the energy used in that contraction is actually lost in the form of heat. So that heat that is generated when our muscles contract is helping us to regulate our body temperature. All right. Questions on this one? Excellent. All right, let's actually do this one on the whiteboard. Wow, that picture is hideous. All righty, new page. We are going to talk about our sensory structures. There's actually only going to be one piece of new information on this, but I want to make sure we understand this information, and so I want us to organize it. What I want us to do is to identify the actual sensory structure. Then we're going to say the type of information it gives us. Then we're going to say the layer or sublayer of the skin where it's found. And we are also going to identify at least one region of the body where we would locate it. So let's go through the ones we have already talked about. One of the ones, the first sensory structure we talked about was that tactile cell. Remember, tactile cells are also called the Merkel cells or the Merkel disc. And what type of information did they give us again?
That was the fine.
There we go. Fine discrimination. Excellent. What layer and or sublayer of the skin did we find these tactile cells? Well, let's start easy. Was it the epidermis or the dermis?
Epidermis.
Is it through the entire epidermis or just one sublayer of the epidermis?
One
sublayer.
The bottom one. And what was that bottom one called?
Stratum basal.
Right, stratum basal or stratum germinativum. Excellent. And while these are found in every region of the body, what were the regions where they were most common?
Hands and feet.
Were these the hands and feet, or were
hands
these
and
hands
the
and
lips.
hands and
Face.
what? Hands and face.
Hands and face.
Excellent. Excellent. Perfect. That wasn't too bad. If I remember correctly, there was another sensory structure that also did fine discrimination. What was that one? Well, I'll give you a start. Same information, so same start of the name. It was a tactile
Corpuscles.
corpuscle. Excellent. Which, remember, we also called the Messner's corpuscle, but again, good old Bob is the one who described that, but you don't need to know that if you don't want to. It also provided fine discrimination, but remember, it was found in a different sublayer and in different regions of the body. What was the layer and sublayer where we found the tactile corpuscles?
It's still in the epidermis.
Not in the epidermis. Excellent. Right. Papillary sublayer of the dermis. Excellent. And while these are found throughout the body, what were the regions where we found the most of these?
Fingertips or palms of your hands.
Yep, hands and feet, right? These were the ones that were on the feet, right? That's why your feet are so sensitive. But notice, hands have a lot of tactile cells and a lot of tactile corpuscles. So what are our most sensitive parts of the body? How do we explore the world?
Hands.
Our hands, exactly. Excellent. Notice this gives us that fine discrimination, but we also had those general sensory information. Those things like tickle, things like itch, things like temperature, things like pain. Those didn't have a special structure at the end. So what was the name for these non-fancy sensory structures called? Yeah, there we go. Free nerve endings. Now remember, this one I mentioned is found in both the dermis and the epidermis. And what parts of your body are capable of feeling pain, capable of itching, capable of measuring temperature?
Everywhere.
Yeah, pretty much everywhere. So not surprisingly, these are pretty much everywhere and in both the dermis and the epidermis. And we're able to receive this basically general sensory information from pretty much everywhere. However, remember the tactile corpuscle wasn't the only corpuscle. There was that layered corpuscle, which is also called the Pacinian corpuscle, because of good old Bob Pacinian. What type of sensory information did that give us again?
Isn't it waterproof? Oh, type of information.
Type. Pressure, absolutely. This is the one that gives us that pressure. Now, if we're feeling pressure, the squeeze of our arm, the squeeze of our back, is this something that needs to be near the surface of our skin?
No.
No. So remember, this is deep in the reticular layer of the dermis. And in some cases, it can actually be in the hypodermis. It is possible to be really, really deep. And again, is there any part of the body where if you squeeze, you're not going to perceive it?
No.
Yeah, so pretty much we see these everywhere as well. Notice there's no new information on the board. This is all stuff we said earlier. I just wanted to organize it so we made sure we understood it. However, there is one more sensory structure I didn't identify for you, and it is called the hair root plexus. And based on its name, the hair root plexus, guess where it's located?
On the hair root.
Yeah, exactly, on the hair bulb. Wait, what? Yeah, it's called the hair root plexus, but it's located around the bulb of the hair. More specifically, around, oops, the follicle that is around the hair bulb. A plexus is an elaborate network of nerves. So it's just this bundle of nerves that is around the bulb of the hair. And what kind of information do you think it gives you?
Movement.
Yeah, exactly. Movement of what?
The hair.
Yeah, exactly. If, and again, I have to work on my analogies because we're not in the classroom, but if a bug landed on your arm, a fly landed on your arm and didn't touch your skin, but just touched your hair, would you be aware of it?
Yes.
If the person next to you, while you weren't paying attention, took their pen and flicked one of the hairs on your arm or grabbed that hair on your arm and pulled it out, would you be aware of that?
Yes.
Yeah, absolutely. So that's what that hair root plexus provides us. And where would you find hair root plexes?
Everywhere but your hands and feet.
Right. And is there a term we could get for that type of skin?
Thin skin.
Thin skin.
Yeah. Yeah, absolutely. Right. So obviously this is anywhere that we have hair. But if you think about it, where the hair is our thin skin. So that's the only one we haven't talked about yet, but I think it's pretty intuitive. And notice, if we look at the pretty picture from your textbook, we can see most of these structures. Notice, as we talked about, here are our free nerve endings, just some nerves that are coming to an ending point, giving us that general sensory information. Notice we don't see a tactile cell. We know it would be here in the epidermis, but notice we do see the tactile corpuscle. We also see the deeper lamellated corpuscle. And notice over here, around not the root, but around the bulb of our hair follicle, we have the hair root plexus. So notice our models show most, not all of these sensory structures. The one sensory structure you need to know about, but you won't identify it on a chart, you won't identify it on a histology slide, is going to be the tactile cells. Notice if we look at our models, notice free nerve endings, tactile corpuscle, lamellated corpuscle. This one doesn't have a free nerve ending. I mean, it probably doesn't have a hair root plexus, but I believe the other one does. Oh, that one doesn't either. All right, well, but luckily we have a drawing. So none of these, it looks like, have that. Could have sworn one of them did. Guess not. All right, but again, that nerve would wrap around the root of the bulb of a hair. So that's the only one we don't have. All right, so there you go. Those are our sensory structures associated with the skin. Questions on that? Like I said, really not any new information there. We have just organized the information we've already talked about. Perfect. Excellent. All right. The last concept we have to cover in class is, now that we understand tissues, we need to talk about how you repair tissues. Now, we are talking about this in the skin because the skin is made up of tissues, but this information is true for all tissues. All tissues basically heal in similar ways because life is lazy that way. Once it finds a system that works, it is going to use that system as many times as possible. So for any tissue to repair, there are certain things that are required. We have to get rid of the dead and damaged cells. We need to bring new cells into that environment to replace the ones that were lost. And are we going to want to do these processes just randomly? Slap stuff in there and hope for the best? No, of course not.
No.
So we are going to need chemical signals to regulate this process. And those chemical signals are either going to come in the form of hormones or some other type of growth factors, right? Like we talked about epidermal growth factor, not a hormone, but it acts just like a hormone. So these are the three things we need to do to repair any type of tissue. Now, do all tissues heal the same way?
No, it depends
No.
on blood supply.
Well, it depends on a lot of things. Yeah, the speed definitely depends on blood supply, but there are other factors as well. Basically, for every tissue in the body, there's one of two ways that they can heal. The first of those healing processes is what we call regeneration. Regeneration is when a tissue is damaged and we replace it with the same type of tissue. So we damage a keratinized stratified squamous epithelial tissue and we replace it with a keratinized stratified squamous epithelial tissue. We damage smooth muscle, we replace it with smooth muscle. And if we're replacing it with the same type of tissue, basically it's going to have the same look and the same function. All right, that makes sense? These are the easy questions, folks.
Yeah.
That makes sense. Excellent.
Yeah.
However, when a tissue heals, it doesn't always regenerate. Sometimes it can heal by a process known as fibrosis. With fibrosis, we are replacing the damaged tissue with a fibrous connective tissue. So let's take that skeletal muscle. I have a shark that takes a big, huge chunk out of my shoulder, and I lose a whole lot of skeletal muscle. Does all that skeletal muscle regenerate back?
No.
No. Instead, it is replaced with a fibrous connective tissue. And is that fibrous connective tissue going to look the same as the muscle that was there before, or function the same as the muscle that was there before?
No.
No. And when that tissue does not look and function the same way, we typically refer to that as scar tissue. And so I would form all this scar tissue that would look and function different from the tissue that was there before. So every single tissue in your body is going to repair in one of these two ways. And how is that decided? Is Monday regeneration day and every other Wednesday is fibrosis day?
No, is it age? How
No.
we do it?
Okay, age could be one of the factors that could influence it. What else could influence it?
How
Mm.
deep the injury is or how severe.
Exactly. The types of tissues that are injured and the severity. If I'm riding my bike and I fall and I scrape my forearm, pretty superficial scrape from a pretty minor injury, which of the two processes is likely to occur, regeneration or fibrosis?
Regeneration.
Regeneration.
Regeneration, absolutely. However, if I take a cheese grater to the entire surface of my arm, is that necessarily, again, I'm not encouraging anybody to do that, but the point I'm trying to emphasize is skin always going to regenerate? No. If instead I take that cheese grater to the entire surface, then you're much more likely to get fibrosis. So some of it is the severity. Some of it is also the types of tissues, right? Like we talked about, skin or epithelial tissues in general are much more likely to regenerate, whereas other tissues, like my skeletal muscle, has limited regeneration. If I'm lifting weights and I, strain the muscle a little bit and injure it, am I going to be able to regenerate that tissue and heal it and maintain function?
No.
Yeah.
Yeah. But like I said, if that shark takes a big, huge chunk out of it, am I going to be able to regenerate all of it?
No.
No. And there are at least two tissues in the body that once they mature, they only heal with fibrosis. They are not capable of regeneration. What are those two tissue types, at least two, that are not able to regenerate when we're adults? I know you know it, even if you don't think you know it. What are some of the organs in the body you don't want to injure?
The liver.
Okay, liver regenerates very slowly. About a cell divides about once a year. Cartilage basically heals via fibrosis because it heals so slowly.
Something with like your spinal cord or your
Nervous tissue. Your nervous tissue. If you damage your nervous tissue even a little bit, does that repair itself?
No.
No. And what's another organ that when damaged doesn't repair itself?
The brain.
Oh, the
Oh,
heart.
the heart.
Heart. There
yeah.
you go.
Cardiac
Heart
tissue.
muscle. Your heart muscle. So cardiac muscle and nervous tissue are ones that basically, once mature, never regenerate, which is one of the reasons why we have to take such good care of them. All right. Are we comfortable with these two repair processes?
Yes.
Did you say cardiac muscle or cardiac tissue?
Muscle.
Okay.
Oh,
Cardiac muscle.
okay.
All right. If we understand these general concepts, then I want to see them in action. And to see them in action, let's actually look at the skin to see some examples of how this stuff will heal.
I have a random question.
Oh, yes.
So like a skin graft, would that count as regeneration? Like if someone, I don't know, has like a severe injury and then they take a graft from like a different part of your location of your body and then place it on there and let it grow.
No, because in the case of a skin graft, you are not growing those cells back. Those cells are being taken from somewhere else. Now,
Okay.
hopefully, if everything goes well, at the edges where it is going to connect to the skin that you have there, hopefully there you will get enough regeneration where you get a minimal amount of scarring as a result of that. So you want to minimize the fibrosis on the edges, but where you just slap a new layer, that area isn't considered regenerated because you've replaced it with different cells. You didn't replace those cells, somebody else replaced them for you.
Okay, thank you.
All right. Let's talk about a superficial wound. Right, with that superficial injury, again, you fall and scrape yourself while riding the bike. And this is one that only includes the epidermis. Now, if this occurs, does your wound bleed?
No.
No, right? Because it's only the epidermis, so there's no bleeding. However, if you look at that scrape, does sometimes some moisture come to the surface?
Yes.
Yeah. Remember, that moisture is that interstitial fluid we talked about. Because you've peeled off some of those superficial layers, the interstitial fluid is coming out. But as you mentioned, because it's only the epidermis, there is no bleeding. Because, oops, where did that go?
Avascular.
There is no bleeding with this type of wound. The dermis is not involved. There is no bleeding. And so in this case, this tissue is going to regenerate itself. Right? Some examples of this are that, oh, hold on. Yes, the basal cells are alive. And in fact, we need those basal cells to be alive to be able to replace the cells that have been damaged. But remember, they're alive, but they don't have a direct blood supply. They're getting their oxygen and nutrients from the dermis underneath. Let's actually look at a pretty picture. Here we see that example. So notice again, down here in the dermis are the blood vessels that are providing the nutrients, providing the oxygen, providing everything for those cells to divide and grow. And as we know, they're going to divide and grow and produce millions of new skin cells every day. But now we have this superficial wound. We have this irritation to the skin. And remember, as we talked about, the damage to the epidermis produces that hormone-like chemical, epidermal growth factor. And in particular, what the epidermal growth factor does is it targets the cells of the stratum basale. It targets those unipotent stem cells of the stratum basale. Now, we know the job of these stratum basale cells is to divide and produce new cells anyway. And that's what they're going to do. They're going to divide to produce new cells. But with epidermal growth factor, which I'm going to go ahead and abbreviate this here because I have it written up before, there's going to be three big differences. They divide faster. They also grow bigger. And not only do they grow bigger, but as they grow bigger, instead of growing up, they actually grow out. And notice we see that here in our illustration. These cells are getting bigger. Oh, that purple doesn't show up well. Let's see if the orange shows up better. No. The cells are growing bigger. And as they're growing bigger, they are also growing outward. Turns out these cells get lonely. When they're damaged and there's nobody next to them, they get lonely. And that's basically what they're doing. They're growing outward looking for a friend. The good news is, as they're growing outward looking for a friend, they're covering up the injury. So as they go bigger and grow outward, they cover the injury. Now notice, like these cells that we see here, eventually the cells from one side are going to reach the cells from the other side. Once they find a friend, do they need to keep growing outward?
No.
No. So now we want them to grow up. And that's exactly what happens. Once they reach another cell, they change the direction of their growth. Right? Instead of growing out, they're going to grow up. Now, of course, as we know, anatomists love to name everything. And so they name this process as well. And this process is called contact inhibition. So when the two cells meet, when they make contact with another cell, it inhibits their outward growth. And now these cells divide and grow upward. And isn't that what our epidermis normally does? It makes new cells. Those new cells make desmosomes that connect themselves together. Then they elongate and they start making proteins, fill themselves up with proteins until they're dead, flattened cells that die and all fuse together. And suddenly, I have replaced the tissue that was damaged with tissue exactly like the tissue that is there. My keratinized stratified squamous epithelial tissue is now replaced with a keratinized stratified squamous epithelial tissue. So again, we have that contact inhibition that stops the outward growth, and then it grows upward, and we regenerate it with the same type of tissue. Looks, feels, and functions the same. And so a week or so later, you can't even tell where that scrape was. Questions on that? All right, that is our superficial wound healing. And notice with a superficial wound, we only use regeneration. However, a deep wound involves the epidermis and the dermis. And so, not surprisingly, it is going to use both healing processes. We already know that the epidermis uses regeneration, but our dermis uses fibrosis. So we are going to see both of these processes. Now, the good news is with a deep wound, the healing of the epidermis is identical. Epidermal growth factors causes our basal cells to grow outward, till contact inhibition, grow upward, replace the tissue. So while I'm sitting here in class explaining this to you, do I have to go through that process again a second time?
No.
No. However, when you're answering this question on the exam, do you want to ignore the epidermis when you're talking about the healing process?
No.
No. If you are asked to describe how a deep wound would heal, make sure you describe that process of regeneration in the epidermis. So again, it is regeneration. Oops. In the epidermis. And we've already done that. So we're going to focus on the dermis and how the dermis heals differently. The dermis uses fibrosis. Notice the other big difference with a dermal injury is we get damaged blood vessels. So this injury bleeds. This damaged blood vessel causes the release of histamine. And as we know, histamine is responsible for our inflammation, redness, swelling, pain, heat, all of that coming on here. And our blood vessels dilate because we want to bring oxygen for healing, nutrients for healing, but we also want to bring the platelets so that we can stop the blood loss. And we want to bring our white blood cells for protection from any harmful pathogens that may come in here. So this area is going to fill up with blood. As it fills up with blood, we know that those fibers that we talked about are going to come out of solution, helping to form that clot. And ultimately, that clot is going to dehydrate, and it is going to form that protective scab on the outer surface of this while it heals. So we start with this inflammation, bringing the blood, bringing all that protection, and stopping the blood loss. And the other advantage of this scab, after the clot forms and the clot becomes a scab, this is going to form a protective cover while the healing takes place underneath. If you get a scab, then yes, blood is involved. So the only way you get a scab is if blood is involved. So that would have to be deep enough to at least get into the papillary layer to cause that scab to form, yes. Notice here, we see that second step, what is now known as the organizational phase. Again, we have this nice scab on top forming this protective outer covering while the healing goes on underneath. Notice also, even though we're not going to talk about it and emphasize it too much, notice our epithelial cells are migrating like we talked about. That epidermal growth factor is having to migrate, try to cover the site of the injury. But like I said, we want to focus on what's going on here inside of our dermis. As we mentioned, a lot of cells come to this area, but there are two types of cells in particular that we get a lot of. We get lots of fibrocytes or fibroblasts. And as we know, what do fibroblasts do? Come on. I know it's late. I know it's been a long day, but we're almost done. What do fibroblasts do?
They create matrix instead of maintaining.
Yeah. And what part of the matrix do you think a fibroblast makes?
Fibers.
Fibers, exactly. They make collagen fibers. We also get lots of macrophages. Macrophages are our big eaters. Their job is to basically break down the dead and damaged tissues and kill any pathogens. And so these two cells become very active. Our macrophages are breaking down all the dead and damaged cells, and the fibroblasts are replacing it with fibers. Now, I don't want to accuse anyone here of being a scab picker, but let's say, for instance, a scab accidentally got scratched off. If you accidentally scratched off a scab and you looked at the tissue underneath, does that tissue look like normal skin?
No.
No, it's very soft and it's very pink and it's very spongy. that special tissue that is forming here in the organization phase under the scab is what is known as granulation tissue. So we're building this granulation tissue and filling up this site of the injury. Now, this is a good thing, but here's the one problem. Here in the skin, there were large glands, there were large hairs, there were large nerves, there were large blood vessels. Are things like the hair, the nerves, the blood vessels, are these the glands? Are these things likely to grow in as fast as this granulation tissue is forming?
No.
So these large structures don't grow back in. And notice that's going to change the function. That's going to change the feel, the appearance. Well, it depends. Some will, but some large ones may not, and they may never return, right? When you get a scar, some scars are there forever. Some scars last for a couple of years and go away. It depends on the severity of the injury. But one of the concerns is, especially in a big gaping wound like we have here, that as this granulation tissue forms, and there's all the words that I just said there, as it's going on in that proliferative phase, we are filling this area so much with fibers that the look, the feel, and the function of that skin can be different. And again, if you think about a scar, if you've ever had a scar, it's typically much more whitish in color because of all the collagen fibers and the lack of large blood vessels. You may not have hairs in that region. You may not have glands in that region. The nerves there could be cut. I have had 3 knee surgeries. On my very first knee surgery, I had my knee reconstructed. It was a 6.5-hour surgery because I'd had some major issues. And there was about a $50 size chunk of skin on the side of my knee that was completely numb. The nerves to it had been cut and I had no sensation from it. And it was probably close to 3 years before I finally got sensation back in that region of my knee. So yeah, is it possible these things could grow back in time? Sure, absolutely. But the problem is we're filling this space with so many fibers, it's going to change the feel. It's going to change the consistency. It's going to change the function of this tissue. Now, I will make one more point. Notice as we've been healing this big wound, it was this big gaping wound, and we're healing it as a big gaping wound. If you have a big gaping wound and you show up at the emergency room, they just go, wow, that's a bummer, and let it heal on its own.
No.
No, what do
No.
they typically do?
Stitch it or glue it.
Yeah, they put stitches in it. And if you put stitches in it, basically you can bring the 2 ends closer together. And as you bring the 2 ends closer together, there's less space in there for this granulation tissue for that scar to form. So by bringing the 2 ends closer together, we can limit, with those stitches, we can limit how extreme that scar is going to be.
Does that mean it speeds up the deep wound healing process faster just because it's a smaller area?
Yeah, it would help with that as well. So probably it's also going to make it less likely that pathogens will get in. So there are multiple benefits to it. Sure. Oh, notice one last thing. Again, I'm not talking about it to emphasize it, but again, notice we've 2 sides of our epidermis have found each other, contact inhibition, and now notice now the layers are growing upwards. Because as all this, well, so when you burn the tissue, the advantage of burning that tissue is you are closing the blood vessels. so that there is less likelihood of blood loss. But you're right, you are putting more damage into the tissue. And so is there more likely that scarring can occur with that cauterization? Yes. So it doesn't stop the process, it just increases the likelihood of scarring occurring. Notice as that fibrosis continues, we reach that maturation stage. Again, notice our entire epidermis is now regenerated, but the dermis is now filled with all of those collagen fibers. At this point, our scab has sloughed off, our epidermis is completely intact and regenerated. And typically those collagen fibers will tighten, and as they tighten and contract, that scar forms. Now notice this one, the skin came basically almost back up to the level, it may be a little below, but for the most part, it tends to respect the boundaries of the skin. However, are there instances that when the healing process occurs, that it can expand out beyond the surface of the skin, forming this bulging scar-like structure?
Yeah.
Yes.
What do we call that condition?
Is it a keloid?
Yeah, keloids. Keloids can form. Why keloids form is not fully understood. What we do know is that they're more common in people that have darker pigmented skin. So the darker the pigmentation, typically the more likely that keloids will occur. The good news is that keloids for the most part are cosmetic, right? Again, it's an enlargement of the scar, but there isn't any other factors. Typically they're not painful or anything like that. In rare instances, there can be some nerves that grow into them and they can be very, very painful. And that can be a problem because is a keloid something that's easy to just go in and cut off?
No.
No, because when you cut it off, what's going to happen?
It's just kind of going to grow back or
Yeah,
grow
the
bigger.
keloid's going to grow back. And some people are hypersensitive to them. They get a mosquito bite and a keloid can form. They scratch their shoulder a little too vigorously and a keloid can form. So for some individuals, these keloids can be quite common. And like I said, we don't fully understand what causes it, but in most cases, for many people, it will eventually respect the boundaries. Like I said, there are just some people that it doesn't. All right, questions on that? All right, we only have a few minutes left, so here's what I want to, here's the things that I'm going to say. Let's do these three things. We only have about 20 minutes left, and I want to use that for histology. So we're gonna do a quick histology review. I know someone earlier mentioned about our normal Q&A review for the exam. We won't have time to do it here, and I don't want to do it here. But what I will do is, as you know, I have my office hours from 12:35 to 1:35. And what I will do is I will hold a review there. So anybody that wants to come and sit and ask questions and listen to questions, we will do it in my Zoom office. Again, it's not required. So for those of you who have other classes or other responsibilities, it won't be required. I'm not going to do it here so that I can close this window and I can start compiling today's lecture so that today's lecture will be available. The third thing, because I want to do the histology review, what we are not going to be able to go over in class, but you are going to be responsible for on the exam. In your book does a nice job of talking about the homeostatic imbalance of the skin, known as burns. For the exam, you need to understand the rule of nines, and you need to understand the degrees of burns, in particular, 1st through 3rd degree burns. When assessing a burn, when you're assessing someone who's been injured with a burn, there are two important things that you have to be able to do very quickly. You have to be able to recognize the severity of the burn, and you need to be able to recognize the extent, how large of a portion of the body is burned. And these are the two features, the degrees and the rules of 9, that help you to assess that. So the rules of 9 is how we, basically it's in simple terms, it's a measure of surface area. So how wide the burn is, and degrees is basically a measure of how deep the burn is. So when someone is burned, that's what we care about. How wide is the burn? How deep is the burn? So your book does a nice job talking about this. I have lecture slides that are on this. You might be able to look back at previous lectures that are on there and be able to get the information from there. I don't want to take the time to go over it now, because instead I would rather we do a quick histology review, because I threw a lot of information at you and I want you to be comfortable with what you're going to be responsible for. All
Are we
right.
also
And then-
responsible for the carcinoma and melanomas?
No, we are not. So great question. We will not do cancers. For skin cancers. So yes, I know that's on there because sometimes we talk about it, but that won't be on the exam. That's too much. But I do want you to know the burns. Okay. All right. Questions on any of that?
No.
All right. Excellent. So let's do this with the time that is left. What I want to do is show you a bunch of slides, have a bunch of arrows, and I will ask the question exactly how I would ask it on the exam. And so your job is to give me the correct answer. See how easy that'll be?
Yes.
Identify the type of skin.
Non-keratinized. Wait. Keratinized.
Identify the
stratified.
type of skin.
Oh, thin.
Thin skin. There you go.
Okay.
Thin skin. This is skin, thin skin. Identify one region of the body where you would find this skin.
Axillary region.
Yeah, axillary, brachial,
Yeah.
perfect, excellent. Identify the layer of the skin. Excellent. Identify the layer of the skin.
Dermis.
Yes.
Dermis, excellent. Identify the specific sublayer.
Stratum cornea.
Excellent. Identify the sublayer.
Stratum granulosa. Is that-
Excellent.
What again?
Identify what?
What was that called?
Stratum granulosum.
Okay.
Identify the sublayer. There we go. Sarah's got it. Stratum spinosum. Identify the, here, let's do it from here. Identify the sublayer.
Stratum basal.
Excellent. Identify the sublayer.
Would that be the
Papillary.
Papillary.
Yeah.
Identify the sublayer.
Reticular.
Excellent. Identify the tissue type.
Keratinized. Stratified squamous epithelium.
Excellent. Identify the tissue type.
Of
Areolar.
Excellent. Identify the tissue type.
Dense irregular.
Excellent. Identify the cell.
Keratinocyte.
Identify the cell.
Keratinocyte.
Identify the cell.
That melanocyte?
That's
Melanocyte.
the melanocyte. Excellent. Um, Identify the membrane.
Mucous membrane.
Mucus?
Oh, yeah.
Cutaneous.
There you go. Excellent. Identify the membrane.
Cutaneous.
Identify the type of skin.
Thick skin.
Identify one region where you would find the skin.
Scales of the foot.
Heel.
There you go, soles. Excellent. Identify the layer.
Keratinized, stratified.
Identify the layer.
Stratum cornea.
Identify the layer.
Epidermis.
Epidermis. Identify the sublayer.
Stratum cornea.
Identify the sublayer.
Stratum
Dermis.
granulosum.
Lucidum. Excellent. Identify the
Oh.
sublayer. Excellent, Sarah. Stratum granulosum. Identify the sublayer. Excellent. Spinosum. Identify the sublayer.
Basal.
Basal. Excellent. Notice, identify the structure.
Papilla.
Papilla. Excellent. Notice in the thick skin, remember we said the papillae are more enhanced. So not only is this structure a papillae, but what sublayer is this?
Dermal papillae.
Well, I would say that is a structure. It's the dermal papillae. But what sublayer is this?
Oh, papillary.
Yeah, papillary sublayer. Excellent. And of course, what type of tissue is it?
Areolar.
Areolar. Notice this one doesn't really show us the reticular layer and the dense irregular connective tissue, so we can't see that well on there. But we can see that. Oh, and again, I guess identify the tissue type. I mean, the cell type.
Keratinocyte.
Yeah, identify the tissue type.
Keratinized squamous epithelial.
Stratified squamous.
Oh, stratified.
Ooh, let's go back a step. Surface.
Apical.
Excellent. Surface.
Basal.
Extracellular material you would find in this location.
Basement membrane.
There you go. See how fun this is and how it all fits together? Awesome. Excellent.
I have a question on the last slide.
Yes.
Did you say the dark purple and the blue larger were both the stratum granulosum?
So the stratum granulosum is this part right here. Right. These are the flat cells, darkly stained, because they are making lots of proteins. Then notice above it is the stratum lucidum.
Oh, okay, thank you.
Notice there is that tiny whitish layer in between, but that's really where the cells are dying and migrating up into the stratum lucidum.
Okay.
Does that make sense?
Yeah.
All right, excellent. All right, so much good stuff here. All right, let's start simple. Identify the general type of gland.
The renal corpuscle.
So again, listen to the question. General type of gland. So this is a gland.
Starts with an S.
It does indeed start with an S and ends in an odoriferous. So what kind of general type of gland is this?
Gland.
Come on, I just gave it to you. What kind of gland is this?
Pseudoriferous.
Pseudoriferous gland. Excellent. Which means what?
Produces sebum.
Do
No,
pseudoriferous glands produce sebum? No, this is going to
That's
produce some kind of sweat. So now that I know it is a pseudoriferous gland, I know two things about it without a doubt. What is its structural classification?
Simple coiled tubular.
Excellent. What is its functional classification?
Merocrine.
Excellent. Now, what specific type of sudoriferous gland is this? Eccrine. Jackie's 100% correct. You guys are correct. How do you know?
The smaller.
The free space and
Yeah, the size of the lumen, the tiny little lumen. So that is a dead giveaway. I'm looking at a sudoriferous gland because it's a simple coiled tubular. And the tiny lumen tells me it is indeed an eccrine sudoriferous gland, which also tells me what it produces. What does this gland produce?
watery sweat.
Watery sweat. And what is the function of the secretion of this gland?
Temperature regulation.
Temperature regulation. And identify a region in the body where you would find this gland.
Anywhere.
Yeah, although don't say anywhere on the exam. Give me one region. And as long as you give me one region, it will be right.
Okay, so
Okay.
we can name any regions and it'll be.
Brachial, antebrachial, popliteal, patellar, femoral, they're everywhere. So give me one region of the body and you have the right answer. Excellent.
So if you ask us for the general gland, that would be the sudoriferous gland. And then if you ask for the specific, it would be the
Eccrine.
eccrine, yeah. Okay.
Notice this one, when I ask for the general type of gland, what's the answer here?
Sudoriferous?
Yeah, notice the general type is the same for both of these. But is the specific answer the same for both of these?
No.
No.
No. Specifically, what type of gland is this?
Eccrine.
Epicrine.
Epicrine.
Absolutely. Structural classification?
The structure, simple, tubular. or
And don't forget coiled, twisted
coiled.
around.
Yes.
Excellent. Although honestly, if you gave me simple tubular on the exam, I'd give you a credit for that. But it's not, doesn't hurt to remember that it's coiled. Mode of secretion or functional classification?
Merocrine.
Merocrine. Excellent. Substance it produces.
Organic sweat.
Organic sweat. Region of the body where you'd find it?
Axillary.
Axillary. Perfect. Buccal, thoracic, anogenital, any of those are acceptable. Again, so much good stuff on this one. All right, excellent. Let's stick with what we've been talking about so far. Right here, identify the specific gland.
Sebaceous?
Exactly, excellent. How do you know?
I just remember because it looks like the fatty ones, but with the nucleus.
It looks like the fatty ones, but it has the nucleus. And notice what it's sitting right next to, some hair follicles. In fact, if you notice,
Where is it?
this hair follicle actually has a hair in it.
Hmm.
So here is a hair follicle. Here is a second hair follicle. And then right next to that hair follicle is a big, huge gland associated with that hair follicle with these big bulbous cells that look like fat, but have nuclei in the middle. So as you mentioned, dead giveaway, this is a sebaceous gland, which is perfect because once we know that, we know everything else about it. What is its structural classification?
Branch. Lobular.
Excellent. What is its functional classification?
Holocrine.
Excellent. Substance it produces.
Sebum.
Function of the stuff it produces.
Moisturizes.
Yeah, moisturizes, antibacterial.
Waterproofs.
Excellent. Region of the body where you'd find it.
Your scalp.
Scalp, axillary, right? Brachial, antebrachial, any place basically where there's hair, where there's thin skin, right? Notice we did the gland, but since the gland is right next to the follicle, we can do that as well. Now we have to cheat a little bit with the hair because we can't really see it. So let's start from the outside of the hair. Notice the easiest part of the hair to see is this dark line on the outer surface. What do you think that dark line on the outer surface is?
Cuticle.
Cuticle, excellent.
Hmm.
So then in here, we'll give a quick little 1 right there. What would that area be?
Medulla.
And then 2 here would be the
Cortex.
cortex, excellent. So that's our hair. Notice this one doesn't even have a hair. So let's jump to this one, because this is our follicle where we have this layer. What do you think that is?
Is that our epithelial tissue?
True, although notice there's 2 of those. So how
Oh,
do
that's
we
the
distinguish them?
internal.
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 Yeah, this is the
Root sheath.
internal epithelial root sheath, which makes the darker purple one what?
The external
External.
epithelial root sheath.
Now notice, remember I talked about how the glassy membrane, I kind of said the follicle has 3.5. Notice this particular follicle, both of these, we don't actually see the glassy membrane. We know the glassy membrane would be here on the outer surface, but neither of these actually show it. So we know it would be there, but neither of these actually show it very well. However, what it does show is the outermost layer of the follicle. And what is this outermost layer of the follicle?
Dermal root. Yeah, sheath.
Yeah, there you go.
Connective tissue.
Exactly, the dermal or connective tissue root sheath. So we have our 3.5 layers of the follicle, our 3 layers of the hair, and that sebaceous gland right next to it. Ooh, any idea what this might be?
Oh,
Yeah, there's the bulb. Exactly. Notice we can see very clearly here. Oops, let's use white. We can very clearly see the medulla and the cortex here. And notice we can see the little bit of that cuticle that is starting to form, right? Just the hint of it at the very beginning here. And notice this does a beautiful job of showing us that papillae, that dermal papillae that sticks there into the center to keep the matrix alive and keep the hair growing. One more fun thing to look at. Any idea what that might be?
Osseous.
corpuscle.
Which corpuscle? Which corpuscle?
A laminated?
Laminated one. Remember, I told you that laminated corpuscle looked like an onion. It really does look like an onion. When this gets squeezed, those layers get stretched, and as they get stretched, we perceive pressure. So, well, lots of things that I say I'm completely making up. Some of the things I say are actually true. And here we actually see that that Pacinian corpuscle, that lamellated corpuscle, really is made up of tons of layers and really does kind of look a little like an onion. All right. I know that was fast, but hopefully that helped to get a sense of what you're expecting for. Ooh,
Yes.
super cool. Wait, one more quick thing. What's this?
Is that adipose?
Yeah,
tissue?
adipose. Notice, like I said, as we run around different tissues, you're going to run into fat everywhere. It is so obvious and so distinct. Again, those connective tissues are super distinct, easy to distinguish. In fact, hold on. I think we only have 3 minutes left in class, but I think that's all we'll need. What connective tissue is this?
Reticular?
Not a bad guess. Has a very cobwebby appearance.
Areolar.
Areolar. Notice we can see really nicely those collagen fibers that are found in here. And notice if we increase the magnification, we can see the darker elastic fibers. And remember, we looked at this slide once before when we were talking about those mast cells containing the histamine. What do we have here?
Adipose tissue,
Excellent, perfect. What do we have here?
reticular,
Excellent. Now, with this reticular connective tissue, what type of fiber is this in the reticular connective tissue?
Reticular fibers.
It's a reticular fiber. What protein is this reticular fiber made up of?
collagen.
Collagen, absolutely, right? Remember that, because when we look at things like our dense irregular, and again, we looked at this in the last class, these are made of collagen as well, but these are collagen fibers. And here we see them at all the different orientations like we would see in the dermis of the skin, whereas here we see all of those collagen fibers lined up like our dense regular. And remember, we also looked at those distinct elongated fibroblast fibrocyte nuclei that make it so obvious and help us to tell the difference between the only two connective tissues that look similar, and that is this one, our dense regular connective tissue, and our elastic connective tissue, where notice we have our dark spot-like nuclei, our coarse fibers. And didn't we say that there was a possible dead giveaway for our elastic connective tissues as well?
The yellow
yellow.
pigmentation.
Yes, sometimes the fibers will be yellow in color. Not all the stains show it this way, but if you see yellow fibers, you don't have to see anything else. You know you're looking at an elastic connective tissue. What's that?
Hyaline cartilage.
Hyaline
Hyaline
cartilage.
cartilage, excellent. Notice this is also hyaline cartilage. We have that nice uniform kind of clear matrix. And remember, we also talked about how it will have that dense irregular connective tissue that wraps around it and attaches it to other tissues. And what did we call this dense irregular connective tissue based on its location? Because it's around the cartilage.
Lamina propria.
That is a locational name, but not this one. This around the cartilage was the
Perichondria.
perichondrium. Exactly, perichondrium. Excellent. Now notice the difference between our matrix here and our matrix here. Right? I got this blown up big on my wall. I got a nice big black light. I grab my bong and my Dave Matthews CD, and I can just stare at this for hours. Right? Look at the difference of the matrix here. These dark, distinct, chaotic fibers going in all sorts of different directions. What type of tissue is this?
Elastic cartilage.
Elastic. This is the elastic cartilage. Absolutely. Remember, fibrocartilage, we see the fibers as well, but notice they're less distinct, and more importantly, the fibers are more parallel with each other. So notice it's a little bit more wavy. They're a little bit more parallel. They're not chaotic all over the place. So for the fibrocartilage, it's much more wavy. And these are collagen fibers, where these are elastic fibers. Notice here, one of the other, again, we were talking about dead giveaways before. One of the things with fibrocartilage is notice they get so many collagen fibers inside of them. Notice they kind of force all the chondrocytes to kind of line up in rows. If you think back to what we looked at in the elastic and the hyaline cartilage, you don't see these rows of chondrocytes. They're kind of randomly spread all over the place. Whereas this has so many fibers to give that compression, that shock absorption. It can actually force the the chondrocytes to line up in rows. Finish easy. What are we looking at here?
Bone tissue.
Osseous tissue.
Excellent. Bone connective tissue or osseous tissue. Identify the space.
Medulla.
Central canal.
Central canal. Identify the space.
Lacunae.
Lacuna.
Identify the cell.
Osteocyte.
Osteocyte. And identify this organizing structure.
Osteon.
Osteon, excellent. And last but not least, identify the tissue type,
Blood.
identify the formed element,
Erythrocyte.
Erythrocyte.
erythrocyte, identify the formed element,
Leukocyte.
leukocyte. Notice there are different types, but you don't have to distinguish the different types. And there's this teeny piece of chewing gum right there. Identify that formed element.
Thrombocyte.
Thrombocyte.
Hmm.
There you go. Took me a couple extra minutes, but again, notice how easy the connective tissues are. They are all so different from each other. All right, so we went a little bit over. Like I said, I'm going to close this window down. If you want to do a Q&A review, then by all means come to my office. The link is on the front page, and I'll happily answer any questions that you have there. But I want to start compiling this so we can get this lecture posted. So any questions before we finish things off? All righty, study hard, study well, and good luck on your exams on Monday.
Thank you.
Thank you.
Thank you.